Part 1. The first dinosaur. Dinosaurs first carved out their niche in the waning days of the Triassic, a period marked by tectonic upheavals and climatic extremes. Fossils such as those attributed to Neosaurus, discovered in Tanzania's Manda beds and dating to roughly 243 million years ago, tantalize with hints of both primitive archosaur and derived dinosaur traits. Its humerus features a pronounced delta pectoral crest, a hallmark of later dinosaurs, yet its overall skeleton retains ancestral features, blurring the line between the first dinosaur and its close relatives. The fragmentary nature of these remains, that delicate crest jutting from a lone arm bone, underscores both the promise and frustration of paleontology's earliest chapter. Tantalizing clues without the complete picture, a story half told in scattered bones and ash-filled riverbeds. Following in its wake, the genus Ankylosaurus, known from coeval Middle Triassic deposits in Tanzania and Zambia, deepens the mystery. Ankylosaurus exhibits upright, columnar limbs and rapid bone growth patterns akin to true dinosaurs, yet it lacks the specialized jaw articulation and pelvic structure definitive of Dinosauria. Its widespread remains across what was once Gondwana suggest a flourishing, diverse group of dinosaur precursors already venturing into varied ecosystems. Each new fragment, whether a partial femur or a single vertebra, poses the question, were these creatures experimental prototypes on the cusp of true dinosaur evolution, or evolutionary side branches that eventually fell away? South America's Ischigualaster Formation, dated to around 231 million years ago, yields two of the most debated candidates, Eoraptor and Herrerasaurus. Eoraptor's slender frame and mixed dentition, sharp, blade-like teeth, paired with leaf-shaped cheek teeth, imply an opportunistic omnivore capable of sampling plants, small vertebrates and insects. Herrerasaurus, somewhat larger and more heavily built, sported recurved, serrated teeth and a robust skull suited to active predation. Phylogenetic analyses flip-flop over their classification. Are they true theropods and sauropodomorphs on opposing branches of the dinosaur family tree, or do they predate that split entirely? Each reinterpretation reshuffles the early dinosaurian deck, demonstrating how unstable the foundation remains. Compounding these uncertainties, fragmentary remains from places as far-flung as Brazil's Santa Maria Formation hint at additional early dinosauriforms yet to be properly described. Partial limbs and isolated vertebrae whisper of species that lived alongside Eoraptor and Herrerasaurus, but whose identities slip through the grasp of researchers. These ghostly echoes suggest a hidden diversity, with distinct lineages experimenting with bipedalism, omnivory, and rapid growth. Pinning down the precise moment when true dinosaurs emerged feels like chasing shadows. Every new bone offers hope, but also deepens the enigma, reminding us how much remains unseen beneath ancient soils. Taphonomic bias deepens the puzzle. While crocline archosaurs and synapsid mammal-like reptiles abound in Triassic deposits, dinosaur bones are remarkably rare. Many scientists surmise that early dinosaurs preferred upland or seasonally dry habitats, areas less prone to rapid burial, where bones would decay or be scattered by scavengers before fossilizing. Conversely, low-lying river valleys favored the preservation of contemporaneous fauna, but may have been ecological backwaters for emergent dinosaurs. This ecological partitioning would explain why the earliest dinosaur fragments appear sporadically in unexpected locations rather than in abundant, continuous layers. Technological advances offer new hope for solving these mysteries. High-precision uranium, lead radiometric dating of ash layers has refined the timing of key fossil horizons by millions of years, reshaping the temporal framework of early dinosaur evolution. Synchrotron CT scans peer inside bone microstructures, revealing growth patterns, vascular networks, and subtle morphological details that distinguish true dinosaurs from near relatives. Isotopic analysis of paleosols reconstructs ancient climates, suggesting seasonal extremes that would have favored rapid growth and endothermy, traits emerging in early dinosaurs. These methods transform fragmentary bones into comprehensive life history records, yet each revelation spawns fresh questions. Ongoing fieldwork in underexplored regions, from the Gobi Desert's remote basins to the karst valleys of southern Africa, continues to unearth critical fossils. Teams endure punishing heat, biting insects, and bureaucratic hurdles, driven by the possibility of discovering a nearly complete skeleton that could bridge the chasm between dinosaur precursors and their descendants. Every round-jawed bone, every tiny claw impression, holds the potential to answer when and where the first dinosaur took its defining step. Erect, bipedal, and destined to dominate terrestrial ecosystems for the next 160 million years. Ultimately, the quest to identify the first dinosaur transcends taxonomic debate. It probes fundamental questions about how major evolutionary transitions occur, how ecosystems recover and diversify after mass extinctions, and how the interplay of anatomy, environment, and chance gives rise to entirely new forms of life. The first dinosaur remains a whisper on the wind of deep time, an unfinished sentence in Earth's grand narrative, awaiting the next fossil discovery to complete its tale. As new layers are uncovered and technologies advance, hope lingers that one day a definitive skeleton will emerge, illuminating that pivotal moment when Dinosauria truly began. Part 2. Feather functions. Feathers first appear in the fossil record among small theropods in the Middle to Late Jurassic, yet their original purpose remains one of paleontology's most beguiling mysteries. Early discoveries of filamentous protofeathers on species like Sinosauropteryx hinted at insulation as a primary function, a surprising trait for scaly reptiles. These delicate hair-like structures likely helped maintain body temperature in fluctuating climates, especially for juveniles with high surface area to volume ratios. Yet insulation alone cannot explain the variety of feather forms discovered, from simple downy tufts to complex pennaceous veins, suggesting that feathers served multiple roles long before the evolution of flight. Among the first puzzles is why some large-bodied dinosaurs, such as Utyrannus, retained extensive feather coverings. At over a ton in weight, Utyrannus challenges the assumption that large animals cannot rely on integumentary insulation without overheating. Its fossils reveal fine filaments coating much of its body, indicating that even apex predators in cooler early Cretaceous latitudes required thermal regulation. How these enormous feathered giants balanced insulation with heat retention and dissipation remains unresolved, especially given the absence of modern analogues at such scales. Feathers also undoubtedly played a role in display and communication. The discovery of iridescent melanosomes in the feathers of Microraptor and Anchiornis suggests that vivid coloration adorned some early pavians. Yet the extent and function of these colorful displays remain speculative. Did these dinosaurs use flash patterns to startle predators, attract mates, or establish social hierarchies? The fossilized patterning tells only part of the story, leaving open the question of how light, movement, and behavioral context combine to create dynamic displays in Mesozoic forests. Another enigmatic function lies in brooding and reproductive behavior. Some oviraptorosaurs, such as Citipati, were found atop nests in adult-like brooding postures, their feathered arms spread protectively over clutches of eggs. Whether these feathers served as thermal blankets, moisture barriers, or simple nest concealment tools remains debated. The lack of direct fossil evidence for juvenile feather coverage means it cannot yet be determined how hatchlings responded to parental brooding. Did they rely solely on egg incubation by ambient heat, or did feather flooding within the nest play a critical role in their earliest survival? Feathers may have also served sensory functions. Modern birds possess phyloplumes, specialized hair-like feathers highly sensitive to tactile stimuli, providing feedback on airflow and feather position. Fossil discoveries of quill knobs on the forelimbs of concavenator hint that non-avian dinosaurs possessed similar structures, yet the distribution, density, and nervous integration of these sensory feathers in extinct species remain unknown. Without fossilized nerve impressions, the extent to which dinosaurs could feel their feathers and adjust them for display, balance, or thermoregulation is an open question. The aerodynamic potential of early feathers, outside of true flight, constitutes another tantalizing mystery. Some non-avian dinosaurs bore long, asymmetrical feathers on their forelimbs and tails, structures capable of generating lift or drag. Microraptor's four-winged planform clearly demonstrates a gliding capability, but the flight functions of other dinosaurs with less obviously aerodynamic feathers, like the ribboned feather bundles of Bipiosaurus, remain speculative. Whether these feathers were ever used for controlled descent, updraft glides between trees, or simple parachuting to break falls invites further biomechanical investigation. Feathers also bear witness to evolutionary experimentation. Multiple lineages developed proto-feathers independently, only to lose them later. The resinosaurids and some ornithomimosaurs, for instance, exhibit evidence of filaments yet lack plumage in derived members. The drivers of feather loss in large-bodied or cold-climate dinosaurs remain unclear. Was it a trade-off between insulation and overheating, a shift in display strategy, or simply genetic drift? The patchy distribution of feathers across the dinosaur family tree underscores the complexity of integumentary evolution and the unsettled nature of feather function beyond the origins of flight. The genetic and developmental pathways that produced feathers in dinosaurs remain largely hypothetical. Comparisons with modern bird embryos reveal the molecular cascades governing feather patterning, but whether these same pathways operated in non-avian dinosaurs and how they varied to produce scales, feathers, or intermediate structures is unresolved. Fossils preserve the outcome, not the genetic blueprint. Without ancient DNA, the deep developmental links between scales, proto-feathers, and feathers remain a captivating frontier, blending paleontology with evolutionary developmental biology in the quest to understand how feathers first took shape on dinosaur skin. Ultimately, the story of feather functions is one of unanswered questions and ongoing discovery. From insulation to display, sensory input to aerodynamic experimentation, feathers opened a world of possibilities for dinosaurs long before flight emerged. Each new fossil find, down to the microscopic arrangement of melanosomes, adds nuance to the narrative while highlighting how much remains unknown. The multifaceted roles of feathers in extinct species stand as a testament to evolution's capacity for innovation, and the mystery of their origins continues to inspire exploration across disciplines. Part 3: Dinosaur Colors. Fossilized feathers and skin impressions have shattered the long-held notion that dinosaurs were uniformly drab, revealing a hidden palette of patterns and hues. Microscopic structures called melanosomes, pigment-containing organelles, preserved in exceptional fossils, illuminate iridescent blacks, rich browns, and even vivid reds and blues. In Microraptor, for example, arrays of oblong eumelanosomes align to create glossy black iridescence across the wing feathers, suggesting displays that flashed with metallic sheen in dappled forest light. Yet the true extent of color variation across Dinosauria remains elusive, as melanosome preservation depends on burial conditions, leaving large gaps in the chromatic record. Beyond black and brown, evidence for pigmentation extends into the realm of reds and yellows. In the wing feathers of Anchiornis, spherical phaeomelanosomes indicate rust-colored patches on the head and wings, hinting at intricate display patterns. Were these colors used in courtship dances, predator deterrence, or interspecies signaling? The exact behavioral context is lost to time, yet the arrangement of colored patches, often in symmetrical arrays, suggests sophisticated visual communication. How these patterns varied between sexes, ages, and seasons remains a compelling mystery, since almost all preserved color data come from mature individuals embedded in fine-grained volcanic ash. In taxa lacking feathers, such as many hadrosaurs and ceratopsians, color evidence is even more scant. Skin impressions reveal scale patterns, polygonal, mosaic-like arrays, but without melanosomes, their hues are unknown. Could the frills of ceratopsians have sported bright ornamentation, flashing warning colors when threatened? Or did hadrosaurs display bold crests and stripes to identify individuals within massive herds? These questions linger because melanosome preservation in scaly skin is extremely rare, requiring exceptional geological circumstances. Until such fossils surface, the coloration of most large dinosaurs remains an open canvas. Camouflage strategies further complicate the picture. Counters hiding dark backs and light bellies has been identified in some small theropods, suggesting a role in obscuring outline under natural lighting. Did large predators like Allosaurus or Tyrannosaurus employ similar patterns to ambush prey in forested or open environments? The absence of direct evidence from skin impressions leaves this possibility speculative, yet the prevalence of counters hiding in modern reptiles and birds implies a likely parallel. How such patterns adapted to varying habitats, woodlands, floodplains, or arid regions across the Mesozoic remains a tantalizing enigma. Seasonal color change, a phenomenon common in modern birds and reptiles, might also have occurred among dinosaurs. Some feathered species could have developed brighter breeding plumages, only to molt into subdued non-breeding tones during other seasons. The timing and mechanism of such molts in dinosaurs are unknown, since fossil specimens capture only single snapshots in time. Without multiple specimens of the same species from different seasons, the dynamics of seasonal color variation remain one of paleontology's most alluring puzzles. Coloration in nestlings and juveniles opens another window into dinosaur life history. Modern birds often display duller hues in their immature stages, gaining vibrant colors as adults. Did dinosaurs follow this pattern? Skull crests in juvenile ceratopsians appear underdeveloped, yet their skin color could have shifted dramatically upon reaching sexual maturity. The developmental cues that triggered such changes, hormonal, environmental, or social, are lost but essential to understanding dinosaur ontogeny. Discoveries of juvenile feathers with preserved melanosomes could illuminate these shifts but remain exceedingly rare. Environmental contexts likely influence color beyond biological need. Dinosaurs inhabiting lush, vegetated floodplains may have favored greens and browns to blend with foliage, while those in open sandy habitats might have evolved earth-tone stripes or blotches for camouflage. Did polar dinosaurs living under extended twilight and snow develop white or pale plumage. Fossils from high-latitude Cretaceous sites, such as those in Antarctica, hold potential answers, but their color data are as yet unexamined. The interplay of habitat, pattern, and predator-prey dynamics in determining dinosaur coloration remains an open frontier of discovery. Finally, the genetic underpinnings of color in dinosaurs are largely speculative. While melanosome structures provide physical evidence of pigment, the regulatory genes and developmental pathways remain inaccessible. Comparisons with modern avian genomes suggest conserved molecular mechanisms for melanin production, but without DNA from fossils, the full genetic architecture is conjectural. The gap between preserved pigment structures and the gene networks that created them highlights a profound mystery. How did dinosaurs deploy and evolve their color palettes over millions of years? The spectrum of dinosaur colors thus remains a vibrant tapestry woven from tantalizing clues and bold inferences. From iridescent blacks glinting on Jurassic wings to imagined patterns on scaly ceratopsian hides, each new melanosome discovery enriches the story while revealing fresh uncertainties. The quest to reconstruct true colors and their meanings continues to inspire paleontologists, blending art and science in pursuit of the world's most ancient rainbows. Part 4. Sound production. Dinosaurs did not rely on silent existence. Their world echoed with low rumbles, resonant booms, and possibly melodic calls that communicated across floodplain forests and open plains. The earliest clues to dinosaurian sound production lie in the elongated nasal passages of ceratopsians such as Corythosaurus and Lumbiosaurus. These crested hadrosaurs possessed complex looping tubes within their skull crests that could function as resonating chambers. CT scans reveal variable chamber sizes and shapes, some narrow and winding, others broad and bulbous, suggesting species-specific calls that may have ranged from trumpet-like blasts to deep throbbing chords. Yet no living analog perfectly matches these structures, leaving the precise timbre and volume of their calls shrouded in mystery. Among theropods, coelurosaurs including Tyrannosaurus rex lacked large cranial resonators, yet their robust hyoid bones hint at a sophisticated vocal apparatus. The presence of enlarged, V-shaped hyoid elements beneath the throat implies powerful control over the glottis and tongue, enabling modulated roars or deep growls. Comparative anatomy with crocodilians, distant relatives of dinosaurs that produce infrasound, suggests that some large theropods could generate low-frequency sounds beyond human hearing. These deep tones might have been used in long-distance communication, territory defense, or mating displays, but direct evidence remains elusive as soft tissues responsible for vocalization rarely fossilize. Ossified thoracic sterna in oviraptorosaurs indicate expanded attachment areas for chest muscles, raising the possibility of air sac-driven calls akin to modern birds. In today's avifauna, spiny tracheal rings and flexible syrinx membranes produce whistles, songs, and trills. The absence of a preserved syrinx in non-avian dinosaurs does not preclude its existence, as the syrinx's delicate cartilaginous tissues seldom fossilize. Fossilized tracheal rings in large sauropods like Apatosaurus exhibit ossified reinforcement, suggesting vocal capabilities that could fill valleys with resonant bass notes. Whether these sounds were haunting moans or rhythmic bellows synchronizing herd movements remains an open question. Vocal sacs, another adaptation in extant frogs and some birds, amplify calls through expandable throat pouches. In Psittacosaurus and some ceratopsians, Paired depressions on the mandible's underside hint at soft tissue attachments that might have supported inflatable air reservoirs. If present, these structures could have functioned to project roars or croaks with minimal energy cost, an invaluable advantage in dense vegetation. Without soft tissue preservation, however, the existence and extent of throat pouches in dinosaurs linger as speculative yet tantalizing possibilities. Hearing structures offer indirect clues to sound production. Inner ear morphology, preserved in some theropod and ornithischian brain cases, reveals sensitivity to specific frequency ranges. In Velociraptor, the semicircular canal dimensions suggest acute low-frequency detection, perhaps tuned to infrasound calls from distant group members. Conversely, smaller herbivores like Protoceratops exhibit cochlear lengths, indicating sensitivity to higher-pitched sounds. These anatomical mismatches imply a complex sonic landscape where species produced and perceived calls tailored to their ecological roles, predators broadcasting deep threats, prey species issuing warning chirps. But confirming these scenarios demands more complete fossilized soft tissues. Trace fossils may yet yield evidence of communication. Fossilized burrows and nests sometimes bear disturbed sediment linings with microscopic wear patterns, as if scuff marked by vocal sac expansions or throat slaps. Similarly, pathologies around nasal openings in some hadrosaurs suggest repeated vibratory strain, possibly from forceful honking. Although such wear could result from other behaviors, the correlation between cranial structures and sedimentary abrasion invites further investigation. Only through meticulous sedimentary fabric analysis can these faint signatures of ancient vocal activity be validated. Ethological parallels with modern animals shed light on unsolved mysteries. In extant crocodilians, subsonic bellows generate ground-transmitted vibrations that travel beneath water or mud. Some hadrosaurs inhabited swampy floodplains. Their nasal crests might have pulsed both airborne calls and substrate vibrations detectable by sensitive feet and jaws. Did these dinosaurs coordinate group movements through seismic signals as well as audible calls? Such dual-mode communication remains hypothetical without direct paleoseismic evidence, making it one of the most intriguing unanswered chapters in dinosaur behavior. Ultimately, dinosaur sound production spans a spectrum from faint trills to earth-shaking roars, mediated by cranial architecture, throat anatomy, and respiratory innovations. The unanswered questions, exact frequencies, call patterns, seasonality, sexual dimorphism in vocalizations, invite a multidisciplinary approach. Combining paleoneurology, sedimentology, digital modeling, and comparisons to living archosaurs, researchers inch closer to reconstructing Mesozoic soundscapes. Yet each new discovery, whether a fossilized tracheal ring or an aberrant crest shape, only deepens the mystery, reminding us that the voices of the dinosaurs still await their full resurrection from the silent stones. Part 5. Warm versus cold blood. The metabolic status of dinosaurs remains one of paleontology's most vibrant debates, hinging on whether these ancient giants were endothermic, maintaining constant internal heat, like modern birds and mammals, or ectothermic, relying chiefly on environmental temperatures. Bone histology provides tantalizing clues. The presence of fibrolamellar bone tissue in many dinosaur clades indicates rapid, sustained growth rates more akin to warm-blooded animals. Blood vessel patterns preserved in cross-sections of sauropod limb bones suggest elevated metabolic demands, yet the sheer size of these giants invites the concept of gigantothermy, a form of inertial homothermy where massive bodies retain heat passively. Determining which dinosaurs actively regulated their temperatures versus those that depended on mass alone remains a complex enigma. Oxygen isotope analyses of tooth enamel and bone phosphate have added another layer to the mystery. By comparing ratios of heavy to light oxygen isotopes in growth layers, researchers infer body temperatures at the time of mineralization. In genera ranging from small theropods to large hadrosaurs, these studies often reveal near constant internal temperatures across seasons, evidence for some level of metabolic heat generation. Yet discrepancies arise. Some sauropods exhibit wider isotopic variation, suggesting partial reliance on ambient warmth. The puzzle deepens when the same technique yields conflicting signals for closely related species, raising questions about ecological, behavioral, or physiological factors that could skew isotopic readings. Feathered theropods offer a direct window into thermoregulation. Protofeathers and downy filaments in species such as Bipiasaurus and Utyrannus point to insulation adaptations, implying a need to retain heat. The dense plumage of the one-ton Utyrannus challenges the notion that large dinosaurs could not overheat while insulated, suggesting behavioral thermoregulation, seeking shade, water baths, or modulating feather orientation. Yet the presence of feathers in smaller Menoreptorans, and possibly even in some ornithischians, suggests insulation evolved early and served critical functions beyond flight origins. Whether insulation's primary driver was endothermy, display, or brooding remains an open question. Growth ring patterns in dinosaur bones further illustrate metabolic diversity. Lines of arrested growth, akin to tree rings, appear in many ectothermic reptiles, but are often subdued or absent in fast-growing, potentially endothermic dinosaurs. In smaller theropods, these rings are rarely observed, hinting at continuous, high-energy growth. Conversely, large sauropods display pronounced growth rings that align with seasonal resource fluctuations, indicating periodic slowdowns. This ontogenetic shift, from rapid endotherm-like juvenile growth to slower adult growth, suggests a flexible metabolic strategy that balanced energy demands across life stages. Yet the exact triggers of these shifts remain enigmatic. Heart anatomy, while unpreserved, is inferred through chest cavity volume and vascular canal density in ribs and vertebrae. Large-bodied dinosaurs likely possessed voluminous hearts capable of pumping blood to extreme body parts, a requirement for sustained activity in endotherms. Crocodilians, as distant relatives, possess a dual-circuit heart capable of shunting blood, whereas birds have a fully four-chambered heart. Whether dinosaurs shared avian cardiac complexity or retained a more primitive system capable of intermittent shunts affects interpretations of their stamina, aerobic capacity, and hunting strategies. The absence of soft tissue fossils leaves this aspect fertile ground for biomechanical modeling and comparative anatomy. Evidence from predator-prey ratios offers ecological context. In modern ecosystems, endothermic predators typically exist at lower population densities than ectothermic ones due to higher energy requirements. Fossil assemblages from the Morrison and Hell Creek formations show relatively low densities of large theropods compared to herbivores, consistent with warm-blooded, high-metabolism predators. However, some Triassic theropods appear more abundant, confusing the pattern. Whether these population structures reflect metabolism or other ecological pressures, such as reproductive rates or climatic stability, remains unresolved, underscoring the need to integrate paleoecological and physiological data. Behavioral inferences also play a role. Dinosaurs likely engaged in behaviors associated with endothermy, active parental care, long-range migrations, and sustained hunting. Migratory trackways attributed to sauropods and hadrosaurs suggest seasonal movements over hundreds of kilometers, an energetically costly strategy more feasible for endotherms. Conversely, large-scale aggregations in some herbivores hint at ectothermic basking behavior, congregating in sunlit areas to thermoregulate. Reconciling these contrasting behaviors within a single metabolic framework challenges paleontologists to consider mosaic strategies, perhaps arranged along a spectrum between cold and warm-blooded extremes. Finally, the concept of mesothermy, a metabolic category between endothermy and ectothermy, is gaining traction. In mesotherms, internal heat production occurs but is limited, resulting in variable body temperatures modulated by both metabolic and environmental factors. Tuna fish and some sharks exhibit mesothermy, using specialized vascular structures for regional heating. Could large dinosaurs have evolved similar adaptations, partial endothermy enhanced by gigantothermy? The discovery of vascularized bone structures capable of heat exchange supports this possibility, offering a middle ground that explains rapid growth and thermal stability without invoking full avian-grade metabolism. The warm-blooded versus cold-blooded debate thus unfolds across bone, feather, behavior, and ecosystem. Each line of evidence, histological, isotopic, anatomical, and ecological, offers tantalizing hints, yet resists a singular narrative. As new analytical techniques refine temperature reconstructions and uncover microanatomical details, the metabolic mosaic of dinosaurs becomes clearer in its complexity and diversity. The enduring mystery of dinosaur thermoregulation stands as a testament to life's ingenuity and the challenges of reading physiology from stone, and promises continued discovery at the intersection of geology, biology, and evolution. Part 6. Sleep behavior. Sleep in dinosaurs emerges as a tapestry woven from trace fossils, rare sleeping postures, and physiological inferences, but the full pattern remains tantalizingly incomplete. One of the most evocative discoveries is a small troodontid preserved in a curled posture, its head tucked beneath its forelimb in a position nearly identical to that of modern birds. This fossil suggests that some non-avian dinosaurs adopted roost-like sleep postures, tucking vulnerable regions away from predators while conserving body heat. Yet whether this behavior was widespread among theropods or unique to certain lineages with avian affinities remains an open question that fossil evidence alone cannot fully answer. Beyond single specimens, trackway patterns hint at communal rest areas. In narrow canyon floors and sheltered floodplain edges, parallel track sets occasionally terminate in clusters of shallow, overlapping prints with no resumption of travel. These sleeping grounds may represent nightly gathering spots where small dinosaurs bedded down together. Whether these gatherings denote flocking behavior for shared vigilance or simply favored microhabitats rich in soft sediment and thus excellent for preserving tracks remains uncertain, but they point to sleep's social dimension among some species. The nesting curves of oviraptorosaurs provide another window into rest and sleep. Adult specimens found atop clutches in brooding postures suggest that parental dinosaurs may have alternated between active incubation and periods of extended stillness, perhaps even sleep intertwined with nest protection. Modern birds shift between warm incubation and shorter feeding breaks. Whether oviraptorosaurs synchronize sleep cycles with incubation shifts or maintained vigilant wakefulness throughout the night is unknown. The absence of nests containing multiple adults leaves the question of communal brooding and alternating sleep schedules tantalizingly unanswered. Some sauropods may have slept standing up, like modern large herbivores. Their massive size would have made rising from a prone position energetically costly and slow, increasing vulnerability to predators. Yet direct evidence is lacking. No preserved sauropod sleeping skeletons have been discovered. Trackways that show groups halting in mid-step, however, hint that these giants paused activity for extended periods, possibly dozing on their feet. How sauropods balanced the need for restful recovery with predator avoidance remains among the most enigmatic aspects of their daily lives. Physiological evidence offers indirect clues. Bone microstructure reveals daily growth increments analogous to tree rings, reflecting metabolic cycles tied to circadian rhythms. Variations in these increments suggest periods of reduced activity, potentially overnight quiescence, common to both endotherms and ectotherms. Yet the precise timing and duration of dinosaur sleep cycles, whether polyphasic like many reptiles or monophasic like most birds and mammals, cannot be resolved without direct behavioral data, leaving a critical gap in our understanding of dinosaur chronobiology. Cranial semicircular canal morphology and inner ear structure shed light on balance during rest. Dinosaurs with specialized vestibular systems, such as agile bipedal theropods, likely required stable posture, even during microsleep episodes. Their ear anatomy suggests rapid arousal capability, enabling split-second reactions to threats, akin to modern prey species that nap with one hemisphere of the brain alert. Whether dinosaurs employed unihemispheric sleep, as some birds and marine mammals do, remains speculative yet intriguing, offering a potential evolutionary bridge between reptilian and avian sleep strategies. Nocturnality itself may have shaped sleep patterns. Evidence from scleral ring dimensions in certain small theropods indicates adaptations for low-light activity, suggesting nighttime foraging and daytime rest. If some dinosaurs were night active, their sleep cycles would invert the diurnal rhythm assumed for most reptiles. The diversity of diel niches, day, night, twilight, among dinosaurs highlights the complexity of reconstructing universal sleep behaviors, as each species likely tuned its rest schedule to ecological and predatory pressures. Finally, thermoregulatory strategies during sleep remain a mystery. Feathered dinosaurs may have fluffed or folded their plumage to conserve heat, much like modern birds roosting in cold climates. Large-bodied dinosaurs with high thermal inertia may have relied on body mass and ambient conditions to remain within survivable temperature ranges while inactive. How dinosaurs modulated blood flow, posture, and integument during sleep to balance energy conservation with predator vigilance is a question that spans anatomy, physiology, and ecology, one that continues to challenge our ability to read behavior from bone. Sleep behavior in dinosaurs thus emerges as a multifaceted enigma, combining fossil snapshots, trace evidence, and functional anatomy. Each discovery, from a curled troodontid to slumbering trackway clusters, adds color to this portrait of Mesozoic nights. Yet the fundamental questions endure. How long did dinosaurs sleep? Where did they choose to rest? And how did their slumber bolster survival in a world of relentless predators and changing climates? Until new fossils or innovative analyses reveal further secrets, the dreams of dinosaurs remain hidden in the silent sediment of deep time. Part 7. Migration Routes. Evidence of seasonal journeys by dinosaurs emerges from the alignment of trackways, bone beds, and chemical signatures locked in tooth enamel, yet the full scope of their migrations remains enigmatic. In North America's Hell Creek Formation, parallel hadrosaur trackways stretching for kilometers suggest long, coordinated movements along ancient river corridors. These footprints often terminate at nesting grounds and water sources, hinting at annual pilgrimages between feeding and breeding areas. Yet the absence of corresponding tracks in intervening regions leaves open the question of whether these paths were used every year or represented episodic mass movements in response to environmental extremes. Isotopic analyses of oxygen and strontium in theropod and sauropod teeth provide a chemical map of prehistoric homelands. Variations in isotope ratios correspond to different geographic regions, revealing that some large herbivores traversed hundreds of kilometers during their lifetimes. In European sauropods, enamel chemistry indicates summer grazing in upland meadows followed by winter retreats to lowland floodplains. How these enormous creatures navigated topographic barriers, mountain chains, and vast swamps without modern landmarks remains a mystery, suggesting they may have followed ancient coastline or river systems no longer preserved. Polar dinosaurs present one of the most striking migration enigmas. In Victoria, Australia, fossils of small ornithopods and theropods appear above the Arctic Circle, where winters plunged into months of near darkness. Did these dinosaurs endure extreme polar conditions through metabolic adaptations, or did they migrate southward during the harshest seasons? The lack of juvenile fossils in polar strata hints at possible nursery sites located elsewhere, but direct evidence of long-distance seasonal movement at high latitudes has yet to be found, leaving their survival strategy one of paleontology's coldest questions. The Prince Creek Formation in northern Alaska offers additional insights into dinosaur migration. Fossil evidence from this region indicates that hadrosaurs, ceratopsians, and theropods may have utilized this area as a migratory corridor between Asia and North America. The discovery of juvenile dinosaur remains in this formation suggests that some species not only migrated through but also nested and raised their young in these high-latitude environments. Trackway analyses have also shed light on the social structures of migrating dinosaur herds. In Denali National Park, the arrangement and spacing of hadrosaur tracks imply organized group movements, possibly indicating coordinated migration patterns. Such evidence points to complex social behaviors among these dinosaurs, akin to the herding dynamics observed in modern ungulates. The debate over whether certain dinosaur species, like Edmontosaurus, were migratory or year-round residents in polar regions continues. While some studies propose that these hadrosaurs undertook extensive migrations, others argue that they remained in high-latitude areas throughout the year, enduring harsh winter conditions. This ongoing discourse underscores the need for further research to unravel the intricacies of dinosaur migration and adaptation. Bone beds containing a mix of juvenile and adult dinosaur remains provide additional context for understanding migratory behaviors. The presence of multiple age groups within these assemblages suggests that entire herds, including young individuals, participated in migrations. This pattern mirrors the migratory practices of contemporary herd animals, where group cohesion offers protection and facilitates the transfer of knowledge across generations. The fossil record also reveals instances of age segregation within dinosaur herds. For example, studies on Hypacrosaurus stebingeri indicate that juveniles formed separate groups from adults until reaching a certain age. Such social structures could have influenced migratory dynamics, with different age cohorts adopting distinct movement patterns or timings. Despite the wealth of evidence supporting dinosaur migration, many questions remain unanswered. The exact routes taken, The triggers for migration and the extent to which environmental factors influence these movements are still subjects of active investigation. As paleontologists continue to uncover and analyze fossil evidence, our understanding of dinosaur migration will undoubtedly evolve, offering deeper insights into the lives of these remarkable creatures. Part 8. Nesting Rituals. Nest building among dinosaurs remains one of the most evocative puzzles of Mesozoic life. Fossilized clutches of eggs, some nestled in neat concentric circles, others scattered as if abandoned in haste, hint at profound behavioral diversity. In Mongolia's Gobi Desert, oviraptorid nests reveal up to a dozen large pear-shaped eggs arranged in tight ovals, each clutch laid and incubated in unison, suggesting synchronized egg-laying events among individuals. Yet the absence of adult skeletons atop many of these nests leaves open the question, did these birds of prey relatives brood like modern birds, or simply cover their nests with vegetation and rely on geothermal heat? The latter strategy would mirror crocodilians, but microscopic pores in some oviraptor eggs hint at an airflow too efficient for burial alone, implying at least intermittent brooding. As paleontologists uncover more nests, the tension between burial and brooding strategies deepens, suggesting that different lineages may have experimented with distinct incubation techniques almost concurrently. Discoveries in the Badlands of Montana have turned the spotlight on hadrosaur nesting colonies spanning hundreds of square meters. Here, hundreds of small, round-tipped eggs lie shockingly close to one another, often in overlapping rings. In Mayasora, dubbed the Good Mother Lizard, fossilized hatchlings and juveniles found alongside nests imply prolonged parental care, perhaps akin to a communal nursery. Yet the degree of interaction between parents and young remains mysterious. Did adult hadrosaurs actively feed their offspring, or did they guide them to food sources and stand sentinel against theropod predators? Isotopic signatures in eggshell carbonate hint at temperature gradients within nests, suggesting careful layering of vegetation to regulate incubation heat. Still, whether adults adjusted nesting material through repeated visits or simply constructed a single thermal mound and departed remains unanswered. Across ancient floodplains of southern Argentina, titanosaur nesting grounds yield hundreds of softball-sized eggs laid en masse. These sauropods, some weighing over 30 tons, undoubtedly lacked the ability to physically sit on their nests. Instead, they appear to have amassed enormous mounds of sediment and plant debris, effectively creating natural incubators. But how these giants coordinated group nesting without trampling nestmates or collapsing incubation mounds is a thorny enigma. Trackways preserved alongside nests show adults skirting the perimeters in tight, organized loops, suggestive of ritualized movement patterns. Could such processions have served both to distribute weight and to signal readiness for egg deposition? And if so, what sensory cues guided sauropods to precise nest sites each breeding season across generations? The geometry of egg arrangement itself poses its own riddles. Many theropod nests exhibit eggs laid with their pointed ends inward, forming perfect radial patterns. This uniform orientation may have optimized heat transfer or facilitated efficient gas exchange, but the exact benefits remain uncertain. In some nests, one or two eggs sit askew, their orientation defying the surrounding symmetry, perhaps indicative of embryonic mortality or last-minute clutch additions. Computed tomography of in situ eggs reveals uneven calcification in such misaligned specimens, suggesting that suboptimal positioning within the nest can impair shell development. Whether adult dinosaurs assessed and corrected errant eggs, or simply accepted reduced hatching success as an acceptable trade-off, is an open question in reproductive strategy. The coloration and patterning of dinosaur eggshells tantalize with possible camouflage functions. Polished fragments from Asian oviraptor nests reveal subtle bands of brown and gray pigments, faint enough to escape detection by human eyes. In ground nesting birds today, cryptic eggs use pigment to blend with substrate, yet non-avian dinosaurs lacked complex color vision, raising questions about selective pressures. Did predatory pterosaurs or small mammals rely on visual cues to locate exposed nests? Or did egg patterning serve thermoregulatory functions, with darker bands absorbing heat more rapidly while lighter regions reflected excess radiation? Laboratory simulations of Triassic daylight conditions suggest that moderate pigmentation could raise incubation temperatures by several degrees, but whether this advantage outweighed predator attraction remains unresolved. One of the most beguiling mysteries surrounds nest site fidelity. Did dinosaurs return to the same nesting grounds year after year? In modern sea turtles and migratory birds, philopatry ensures that hatchlings imprint on proven locales. Fossil nests in Patagonia, however, show variations in sediment composition and depth across successive layers, hinting that some dinosaur species shifted nesting sites across generations. Yet isolated rows of trackways leading from distant feeding grounds to long-abandoned nesting areas imply occasional, ritualistic return journeys. How these animals imprinted on nesting territories by olfactory, magnetic, or visual landmarks, and how they communicated these locations to their offspring, remain speculation. Egg-turning behavior is integral to modern avian incubation, preventing embryo adhesion to shell membranes and promoting uniform development. But for non-avian dinosaurs, direct evidence of such behavior is scant. Some oviraptorid nests preserve depressions in the surrounding sediment, shallow gouges that could represent beak-driven egg adjustments. Alternatively, these marks might be artifacts of nest construction or even trampling. Experimental modeling of nest thermodynamics shows that unattended static clutches suffer from thermal stratification, implying a strong selective pressure for egg turning. Whether dinosaur parents turned eggs with their snouts, feet, or even body shifts is a profound unknown, one that may be illuminated only by fortuitous preservation of articulated skeletons atop intact clutches. Embryonic pathologies reveal another layer of puzzle. Flattened, crushed, or maldeveloped eggs are not uncommon in fossil beds, suggesting environmental stresses or nutritional deficiencies. In one notorious troodontid nest, over half the clutch exhibits abnormal shell thickening. This could indicate high mineral content in local water sources, or perhaps a genetic predisposition toward shell malformation under extreme temperatures. The uneven distribution of such pathologies across taxa and locales points to complex interactions between diet, climate, and reproductive physiology. Yet pinpointing the primary culprit among these factors remains elusive, with no two nesting sites presenting an identical pathological signature. Finally, the broader question lingers: how did evolving nesting rituals shape dinosaur diversification? Species that developed efficient brooding and hatchling care may have enjoyed higher reproductive success, fueling the explosion of new forms. Conversely, those that relied on simple burial may have been more vulnerable to climatic upheavals and habitat changes. As paleontologists continue to unearth new nesting grounds, from the arid badlands of North America to the volcanic ash-strewn plains of China, the layering of ecological, anatomical, and behavioral data gradually coalesces into a richer portrait. Yet until a perfectly preserved dinosaur parent is discovered in life position guarding its nest, the true choreography of these ancient rituals will remain one of paleontology's most elegant enigmas. Part 9. Hatchling Survival. The moment a dinosaur cracked its way out of the egg marked a culmination of a perilous journey. Fossilized nests of the hadrosaurid Maiasaura expose a remarkable scene. Dozens of tiny hatchlings, each scarcely the size of a chicken, crowded together in a shallow depression lined with broken eggshells. Their long, gracile forelimbs and unfused ankle bones reveal that they were not yet strong enough to walk long distances, making the nest a literal cradle of vulnerability. Paleontologists have found delicate tooth marks on some eggshell fragments, evidence that small carnivorous lizards and mammals seized hatchlings before they could fully emerge, an ominous reminder that dinosaur infancy was beset by constant predation. Yet the clustered arrangement also suggests a communal defense strategy, with dozens of young calling out in unison. The chorus may have deterred solitary hunters, though it likely also broadcasted their location to larger theropods waiting in ambush. Bone histology from juvenile Maiasaura shows rapid growth rates in the first few months of life, implying that hatchlings needed to bulk up quickly to outgrow the predator window when they were small enough to be hunted but too large to hide effectively. Lines of arrested growth, lags preserved in limb bones, indicate that stress episodes, periods of slowed growth, occurred regularly during early ontogeny, perhaps linked to seasonal fluctuations in food availability or climatic stressors like drought. Yet the precise triggers for these pauses remain obscure. Did rainfall patterns in the late Cretaceous dictate brooding success? Or did dietary shifts from soft leafy shoots to tougher ferns impose metabolic bottlenecks? Until stable isotope profiles from multiple nesting sites are compared, the relative influence of environment versus physiology on hatchling survival will remain unsettled. Troodontid hatchlings, by contrast, appear to have been far more precocial. Tiny skeletons with well-ossified limb girdles and open eye sockets suggest that they could move swiftly and forage independently almost immediately after hatching. CT scans of embryonic troodontid skulls reveal enlarged semicircular canals, inner ear structures crucial for balance, pointing to advanced vestibular development before birth. Yet how these miniature predators avoided competition and cannibalism within their own species is perplexing. Their nests, often found in arid deposits, contain fewer eggs and are more widely spaced, hinting at a solitary strategy rather than communal nesting. Did troodontid parents actively teach young hunting techniques, or were hatchlings left to their own devices, relying on innate instincts honed by millennia of evolutionary pressure? Among oviraptorids, the discovery of embryonic remains, with well-formed limb joints inside eggs, suggests a unique model of sit-and-wait survival. Hatchlings may have been capable of rapid sprinting to escape danger soon after pipping. Yet the depictions of brooding adults found atop nests have led some researchers to propose that parent oviraptorids guarded their nests post-hatching as well, a behavior unseen in most modern reptiles. If true, this would require a physiological capacity for endurance covering weeks of nest attendance without feeding, a metabolic feat that raises questions about adult dietary adaptations and energy reserves. Microscopic wear patterns on fossilized teeth of presumed brooding adults hint at a specialized diet of shellfish or other high-protein sources, possibly consumed near nesting sites to sustain non-incubating adults. Clutch size directly influenced hatchling competition. In smaller-bodied dinosaurs like Protoceratops, clutches rarely exceeded a dozen eggs. Yet juvenile bone beds sometimes contain dozens of half-grown individuals jumbled together, suggesting that in some cases multiple broods coalesced into nursery herds. Did these aggregations result from synchronous hatching in overlapping territories, or did older juveniles join younger ones in search of safety in numbers? Furthermore, growth ring analyses show that within such groups, size disparities could be substantial, implying that hatching asynchrony forced late-emerging siblings into subordinate niches. Whether dominant juveniles monopolized prime foraging spots while stragglers faced malnutrition, or whether all young shared resources communally, remains an open debate. Disease and injury also shaped hatchling fate. Fossilized limb bones of some young sauropods show signs of osteomyelitis bone infections, likely resulting from wounds sustained during nest excavation or early life struggles. Other specimens exhibit healed fractures, suggesting that hatchlings somehow survived serious accidents long enough to regenerate bone tissue. How these infants coped with infection before their immune systems matured is unknown. Did maternal antibodies transfer through eggshell membranes, analogous to antibodies found in modern birds? Preliminary biochemical analyses of eggshell proteins hint at the presence of glycoproteins potentially involved in antimicrobial defense, but the extent and efficacy of such natural antibiotics in dinosaur eggs remain speculative. The ambient environment posed its own threat. In the arid lowlands of late Cretaceous Mongolia, extreme temperature swings could bake nests by day and frost them by night. Sedimentary evidence indicates that some nests were submerged by flash floods, scattering eggs and hatchlings alike. Yet clutch relocations documented at certain hadrosaur sites, where eggshell fragments lie far from adult trackways, suggest that some dinosaurs may have rescued inundated nests, carrying eggs to higher ground with their beaks or forelimbs. Were these salvage operations instinctual responses to nest disasters, or did they form part of a broader repertoire of parental care? The lack of adult skeletal remains associated directly with moved clutches leaves this question tantalizingly unresolved. Dietary transitions in hatchlings remain shrouded in mystery. Carbon isotope ratios measured in the first growth rings of juvenile hadrosaur teeth differ markedly from those in later rings, indicating a shift from a milk-like fluid produced in specialized crop-like organs to a diet of fibrous plants. Crop milk is known in pigeons and flamingos today, but evidence for an analogous system in dinosaurs derives primarily from gut content fossils of adult oviraptorids, where partially fermented plant matter suggests foregut fermentation. Whether duck-billed dinosaurs and possibly other lineages secreted nutritive fluids for their young, and how long this provisioning lasted, are questions still waiting for that rare moment-in-time fossil capturing a hatchling in the act of suckling or drinking. Ultimately, hatchling survival was a high-stakes game of numbers and strategies. Some species may have relied on sheer reproductive output, hundreds of eggs per season, to ensure that a few survivors reached adulthood. Others, like Myosaura, invested heavily in a smaller number of offspring, nurturing them through extended care. The evolutionary trade-offs between many eggs, little care, and few eggs, intense care, likely influenced dinosaur life histories and extinction vulnerabilities. Until we unearth more exquisitely preserved juvenile-adult associations and embryonic tissues revealing physiological data, the myriad tactics dinosaurs employed to shepherd their young from fragile hatchling to robust juvenile will continue to weave one of paleontology's most compelling enigmas. Part 10. Herd Dynamics. Fossil quarries in Montana and Alberta have exposed sprawling mold fittings of hadrosaur bones, hundreds of individuals packed together after catastrophe. These bone beds, dominated by Maiasaura and its lambeosaurine kin, reveal age-segregated assemblages in which juveniles, subadults, and adults lie in distinct clusters. Such size sorting implies that herds were structured by age, perhaps reducing intraspecific competition for food while maximizing collective vigilance against predators. Yet the precise social rules governing these groupings elude researchers. Did hatchlings join creches under the watchful eyes of subadults before rejoining the main herd? Or did adults shepherd cohorts of different ages through seasonal migrations, reuniting each year at traditional calving grounds? Without articulated trackways showing mixed-age stride patterns, the choreography of these journeys remains a tantalizing blank in the record. In Asia's Gobi Desert, oviraptorid bone beds demonstrate a different mode of herding. Here, smaller flocks of perhaps a dozen animals cluster together, their nests often sited within meters of one another. Eggshell fragments, intermingled with adult bones, suggest communal nesting colonies, a strategy that trades individual secrecy for shared defense. Yet the limited clutch sizes, far smaller than those of sauropods or hadrosaurs, suggest that oviraptorid herds relied heavily on social cohesion rather than sheer numbers. How these flocks maintained harmony and whether dominant individuals enforced spacing or nest hierarchies remains unknown. The absence of nest-guarding trace fossils compounds the mystery. Did some members stand sentinel while others incubated, or did incubation itself become a rotation of duties? Ceratopsians add another dimension to herd dynamics. Massive bone beds of Centrosaurus and Styracosaurus preserve herds numbering in the hundreds, with individuals all facing in the same direction, evidence of catastrophic die-offs at watering holes. Sedimentology indicates these herds were mobile and lived in open floodplain environments, but the evolutionary drivers of their gigantism and numbers remain unclear. Did horns and frills facilitate individual recognition within vast herds, helping to prevent inbreeding and coordinate group movements? Some hypotheses propose that frill shape and horn arrangement served as barcode signals, yet no trackways or footprints conclusively link specific frill morphologies to herd movement patterns. The possibility of visual signals coordinating million-strong migrations remains one of paleontology's grand questions. Evidence from South America pushes herd living back even further. Mosaurus, a relatively small sauropodomorph dating to roughly 193 million years ago, appears to have nested and possibly migrated in groups of juveniles and adults alike. Losic silt deposits in Patagonia preserve dozens of skeletons at various ontogenetic stages together, hinting at age-segregated nursery herds. Isotope ratios measured in teeth of juvenile and adult Mosaurus from the same site reveal distinct feeding habitats, suggesting that young sauropods ranged separately before rejoining adults, much like modern elephant herds. But the trigger for these separations, whether food scarcity, predation risk, or social learning, remains debated. Until more isotopic datasets emerge, the drivers of age-structured herd fusion dynamics will stay unresolved. Not all dinosaurs joined large herds. Ankylosaurs seem conspicuously absent from communal bone beds, suggesting a more solitary or pair-bonded lifestyle. Their heavy armor and clubbed tails imply defensive reliance on morphology rather than group vigilance. Yet isolated trackways occasionally show 2 or 3 ankylosaurs moving in parallel, raising the possibility of small family groups. Without bone beds of dozens of ankylosaurs, the evidence is circumstantial and the question of whether they foraged alone, in pairs, or in tiny clans remains unanswered. If small group living existed, what ecological pressures favored it over massive herd forming, a mystery that challenges assumptions about social evolution among armored dinosaurs. Predator avoidance likely drove the evolution of herding in many herbivorous dinosaurs. Trackway studies show mixed cohorts of hadrosaurs and ceratopsians moving together in the two-medicine formation, possibly suggesting multi-species herding for increased detection of theropod threats. Such safety in numbers might have required complex interspecies communication. Did different herbivores share alarm calls, communicated through low-frequency vibrations like modern elephants? Fossilized bone beds seldom preserve soft tissue structures for sound production, leaving open the question of whether these giants could coordinate across species boundaries in real time. Seasonal migrations introduce further complexity. Oxygen isotope analyses in hadrosaur bones from high-latitude sites indicate annual movements between coastal lowlands and inland highlands to exploit seasonal plant growth. But it remains unknown how herds navigated such long distances. Did they follow ancestral routes imprinted in memory, or did each generation learn by following elders? The lack of continuous graded trackways spanning hundreds of kilometers in any one site leaves researchers guessing at the cues, magnetic, olfactory, or visual, used by migrating dinosaurs. This gap in understanding blurs the picture of herd coordination over the landscapes of Cretaceous continents. The emergence of herd-based age segregation also influenced growth and survival strategies. In species like Maiasaura, flowing data from bone histology shows that juveniles grew faster within nursery herds, possibly due to cooperative foraging and shared parenting. Yet not all herding dinosaurs display this pattern. Some ceratopsians reveal similar growth rates across all age classes, suggesting uniform resource distribution. Whether this reflects differences in metabolic demands, social structures, or habitat resource availability remains unresolved. Future quantitative modeling of herd density versus growth rates may untangle these variables, but for now the interplay of physiology and sociality in dinosaur herds endures as a deep mystery. Finally, the evolutionary cost-benefit balance of herding continues to intrigue. Large herds offered protection and improved breeding success, but also risked mass mortality during environmental catastrophes, floods, droughts, or volcanic ash falls. Bone beds frequently capture these tragedies in geological snapshots, yet the prevalence of herd-forming lineages into the very end of the Mesozoic suggests that, on balance, herding paid off. How herd dynamics shifted in the final million years before extinction, and whether social behavior altered dinosaurs' vulnerability to rapid climate change, remains one of the last unsolved chapters in their long saga. Part 11. Sexual dimorphism. In the fossilized remains of the late Jurassic stegosaur, Hesperosaurus majosi, researchers have identified two distinct plate morphologies, one featuring broad oval-shaped plates and the other displaying taller, narrower forms. These differences are not attributable to age or species variation, suggesting they may represent sexual dimorphism. The broader plates could have served as visual displays, possibly for attracting mates or deterring rivals. However, without definitive evidence linking these plate types to specific sexes, the exact nature of this dimorphism remains speculative. The hadrosaurid Maiasaura peebalzorum presents another intriguing case. Statistical analyses of its fossil record indicate a significant size disparity between individuals, with one sex being approximately 45% larger than the other. This pronounced difference suggests sexual dimorphism, but determining which sex was larger is challenging. In modern animals, larger size can be associated with either males or females, depending on reproductive roles and behaviors. Without soft tissue preservation or definitive sex indicators, assigning these size differences to a specific sex in Maiasaura is currently not possible. Protoceratops Andrew C., a ceratopsian from the late Cretaceous, exhibits variations in frill and nasal prominence sizes among individuals. Some paleontologists interpret these differences as evidence of sexual dimorphism, with larger frills possibly serving as display structures for mate attraction or dominance displays. However, others argue that these variations could result from age-related changes or individual variation rather than sex differences. The lack of clear, consistent patterns makes it difficult to confirm sexual dimorphism in this species. In the early Jurassic theropod Coelophysis bari, two distinct morphs have been identified: a gracile form with a longer skull and neck, and a more robust form with a shorter skull and longer arms. Initially, these differences were thought to represent sexual dimorphism, with the robust form being male. However, further studies suggest that these variations may be due to individual growth differences rather than sex. The high variability in growth patterns among Coelophysis individuals complicates the interpretation of these morphs as sexually dimorphic. The oviraptorid Cytipati osmolskae provides compelling evidence of parental care, with fossils found in brooding positions over nests. While this behavior indicates a level of reproductive investment, determining the sex of these individuals is challenging. If both sexes participated in nest guarding, it could suggest a more equitable division of parental duties. However, without clear sexual dimorphism in skeletal features, it's difficult to ascertain whether these behaviors were sex-specific or shared. In the case of the heterodontosaurid Ebrictosaurus consus, the presence or absence of caniniform teeth has been proposed as a sexually dimorphic trait. Some specimens possess prominent tusks, while others lack them. This variation could indicate differences between males and females, similar to tusk development in modern mammals. However, the possibility that tusk absence is due to juvenile status rather than sex complicates this interpretation. Without more comprehensive data, the role of tusks in sexual dimorphism for this species remains uncertain. The sauropod dinosaurs, such as Apatosaurus and Diplodocus, have been hypothesized to exhibit sexual dimorphism in tail vertebrae structure. Some specimens show fused caudal vertebrae, potentially allowing for tail elevation during mating. This feature is present in only a subset of individuals, suggesting a possible sex-related difference. However, the correlation between vertebral fusion and sex is not definitively established, and alternative explanations, such as individual variation or developmental differences, cannot be ruled out. In theropods like Tyrannosaurus rex, attempts to identify sexual dimorphism have focused on skeletal robustness and pelvic morphology. Some studies suggest that females may have had wider hips to accommodate egg-laying. However, the overlap in size and shape between individuals makes it difficult to distinguish sexes based solely on these features. The lack of consistent distinguishing characteristics in the fossil record limits our ability to confirm sexual dimorphism in these large predators. Overall, the study of sexual dimorphism in dinosaurs is fraught with challenges due to the incomplete nature of the fossil record and the absence of soft tissue preservation. While certain morphological differences among individuals suggest the presence of sex-based traits, definitive conclusions are elusive. The interplay of growth stages, individual variation, and potential dimorphic features creates a complex puzzle that continues to intrigue paleontologists. As new discoveries emerge and analytical techniques advance, our understanding of sexual dimorphism in these ancient creatures may become clearer, shedding light on their reproductive strategies and social behaviors. Part 12. Social structures. In the fossil-rich expanses of Patagonia, the discovery of Mosasaurus patagonicus assemblages, dating back 193 million years, reveals compelling evidence of complex social organization among early sauropodomorphs. These assemblages include nests, eggs, juveniles, and adults, suggesting that these dinosaurs lived in structured communities with age-segregated groupings. The presence of such organized social behavior in early dinosaurs challenges previous assumptions and indicates that sophisticated social structures may have been a factor in their evolutionary success. Further north, in the Two Medicine Formation of Montana, extensive bone beds of Maiasaura peeblesaurum provide additional insights into dinosaur sociality. These sites contain evidence of nesting colonies with adults, juveniles, and hatchlings coexisting, suggesting that Maiasaura engaged in communal nesting and possibly cooperative care of the young. The arrangement of nests and the developmental stages of the juveniles imply a level of parental investment and social coordination uncommon among reptiles, aligning more closely with behaviors observed in some modern bird species. In Inner Mongolia, the discovery of a group of Sinornithomimus dongi individuals, all juveniles, preserved together in what appears to be a mud trap, offers a snapshot of social behavior among ornithomimids. The age uniformity of the group suggests that these dinosaurs may have formed age-specific cohorts, potentially for foraging or predator avoidance. The absence of adults in the assemblage raises questions about the social dynamics of these groups and whether juveniles operated independently or under the guidance of adults. The ceratopsian Protoceratops andrewsi, known from the late Cretaceous of Mongolia, provides intriguing evidence of social interactions. Fossil sites have yielded multiple individuals preserved together, including a famous specimen locked in combat with a velociraptor. The frequent association of Protoceratops individuals suggests gregarious behavior, and the presence of varying frill sizes among individuals may indicate social hierarchies or sexual dimorphism. However, the exact nature of their social structures remains a subject of ongoing research. In the realm of theropods, the discovery of trackways attributed to dromaeosaurs in Shandong Province, China, provides rare evidence of potential group behavior among these predators. The parallel trackways suggest that these dinosaurs moved together, possibly indicating coordinated hunting or social movement. This challenges the traditional view of theropods as solitary hunters and opens the possibility that some species exhibited complex social behaviors akin to pack hunting. The massive bone beds of Centrosaurus apatus in Alberta, Canada, offer a glimpse into the social lives of ceratopsians. These sites contain thousands of individuals preserved together, suggesting that Centrosaurus lived in large herds. The uniformity in age distribution within these assemblages implies that these herds may have been structured by age, with potential implications for social organization, mating systems, and predator defense strategies. Sauropod trackways from various global locations, including those attributed to Alamosaurus and Belosaurus, reveal patterns of movement that suggest herding behavior. Some trackways indicate age-segregated groups, while others show mixed-age herds, reflecting diversity in social structures among sauropods. The reasons behind these differing herd compositions remain unclear, but they may relate to ecological factors, predation pressures, or reproductive strategies. The presence of communal nesting sites among various dinosaur species, such as those found in India and Argentina, further supports the idea of complex social behaviors. These sites often contain numerous nests in close proximity, indicating that dinosaurs return to specific locations to lay eggs, possibly in synchronized events. Such behavior suggests a level of social coordination and site fidelity that parallels nesting behaviors observed in some modern bird species. Despite the growing body of evidence for social behavior in dinosaurs, many questions remain unanswered. The extent to which dinosaurs engaged in cooperative behaviors, established social hierarchies, or exhibited division of labor within groups is still largely speculative. Future discoveries and advances in technology may provide further insights into the social structures of these ancient creatures, shedding light on the evolutionary origins of sociality in vertebrates. Part 13. Brain Size Mystery. Among the most enduring enigmas in paleontology is the disparity between the immense bodies of certain dinosaurs and their comparatively diminutive brains. Take, for instance, the sauropod Ankylosaurus. Despite its towering stature, its brain was scarcely larger than a walnut. This disproportion raises questions about how such colossal creatures managed their bodily functions and behaviors with such limited neural resources. The mystery deepens when considering that brain tissue rarely fossilizes, leaving scientists to infer brain size and structure from cranial cavities, which may not have been entirely filled by the brain itself. The result is a murky window into cognitive capabilities that may have differed drastically from what skull volume alone would suggest. The myth of dinosaurs possessing a second brain in their hips, particularly in species like Stegosaurus, has long been dispelled. This misconception arose from an enlarged neural canal in the sacral region, which some early paleontologists misinterpreted as a supplementary brain meant to control the massive hind limbs and tail. In reality, this space likely housed a glycogen body, an energy-storing structure also found in modern birds. Although not a second brain, this adaptation may have played a role in locomotor efficiency or balance, but it adds a layer of intrigue to how such anatomical features were misunderstood for generations. Contrasting the small-brained giants, certain theropods like Tyrannosaurus rex present a more complex picture. Some reconstructions of its brain case suggest that T. rex may have had more cognitive potential than previously assumed. The possibility that this apex predator had a brain dense with neurons invites speculation about its hunting strategies, problem-solving skills, and even social behavior. However, interpretations of intelligence based on brain volume and neuron estimates remain hotly debated. Without direct behavioral evidence, the notion of a clever T. rex teeters between possibility and wishful thinking, leaving researchers sifting through fragments of bone and inference. Hadrosaurs, or duck-billed dinosaurs, often associated with herd behavior and vocal communication, provide an interesting counterpoint. Endocasts of species like Edmontosaurus reveal relatively large cerebral hemispheres, especially in comparison to their herbivorous peers. Their encephalization quotients, measures of brain size relative to body mass, suggest higher than average neural investment for a plant eater. Whether this translated to sophisticated social interactions, enhanced memory, or greater sensory processing remains a tantalizing mystery, but it complicates the simplistic view that braininess belonged solely to carnivores. An extraordinary fossil from Sussex, England, preserved what is possibly the first fragment of fossilized dinosaur brain tissue ever discovered. The delicate mineralized remains, which include fine details of meninges and blood vessels, offer an unprecedented glimpse at the soft anatomy hidden within skulls. This rare preservation suggests a brain structure more complex than previously imagined for the dinosaur it belonged to, likely a small ornithopod. The find raises new questions about how often such preservation might have occurred without being recognized, and what other secrets might lie dormant in overlooked fossils. Troodon, a small theropod known for its large eyes and advanced sensory capabilities, has long been considered one of the most intelligent non-avian dinosaurs. Its unusually high brain-to-body size ratio has led to speculative reconstructions imagining its evolutionary trajectory had the mass extinction not intervened. Some have even proposed that Troodon could have developed complex societies or tool use over time. While such projections border on science fiction, they illustrate the compelling idea that intelligence was not unique to mammals or birds, but perhaps simmered in the background of the Mesozoic for millions of years. Brain size alone, of course, does not dictate behavior. Some dinosaurs with modest brains may have exhibited surprisingly complex actions through hardwired instincts and specialized brain regions. Others with relatively larger brains may have used them for processing sensory input or coordinating movement rather than abstract thought. The diversity of dinosaur neuroanatomy complicates any one-size-fits-all interpretation of intelligence and reminds researchers that evolution tailors cognition to ecological niche rather than conforming to linear progression. Modern imaging technologies like high-resolution CT scans have allowed paleontologists to peer inside skulls with unprecedented clarity. These scans produce digital endocasts that reveal the shape of the brain cavity, nerve pathways, and vascular networks. They have helped clarify distinctions between different parts of the brain, such as olfactory bulbs and optic lobes, shedding light on which senses were most important to specific dinosaurs. Yet despite these advances, translating anatomical data into behavioral insight remains a leap, one that continues to inspire new methodologies and frameworks. Ultimately, the mystery of dinosaur brain size underscores the broader challenge of reconstructing cognition in extinct animals. Without direct behavioral observations, scientists must rely on comparative anatomy, biomechanical modeling, and ecological inference to piece together the mental worlds of creatures lost to time. As new fossils are uncovered and techniques refined, the story of dinosaur intelligence may evolve into one of the most surprising subplots in the grand narrative of prehistoric life. One where brains, not just bones, shape the legacy of the Mesozoic. Part 14. Growth rate variance. Dinosaurs exhibited an astonishing range of growth rates, from fast-growing theropods to slower-developing armored species, revealing complex biological strategies that remain only partly understood. Examining their fossilized bones, especially the microscopic structure, reveals how different lineages grew at vastly different speeds. Some species laid down layers of fibrolamellar bone, indicating rapid, sustained growth, while others showed evidence of pauses or slowdowns, visible as growth rings akin to those in trees. These patterns offer insights not just into how quickly a dinosaur matured, but how it adapted to its environment, reproduced and competed within its ecosystem. Few dinosaurs illustrate dramatic growth spurts better than Tyrannosaurus rex. Juveniles may have resembled lanky, fast-moving predators before undergoing an adolescent explosion in size. Studies suggest that between ages 14 and 18, a T-Rex could gain up to 600 kilograms per year, ballooning into the apex predator it's remembered as. This explosive growth may have helped juveniles avoid direct competition with adults by occupying different ecological niches, a rare example of age-based resource partitioning in extinct species. The mystery, however, lies in what triggered this shift and whether it was hormonally or environmentally driven. In contrast, the sauropods, giant long-necked herbivores like Apatosaurus, appear to have matured at breakneck speed, reaching adult sizes in as little as 15 years. Their colossal mass and massive bones demanded a physiology capable of rapid construction. Histological evidence shows dense networks of blood vessels and growth rings that often blur together due to the speed of deposition. This fast growth may have served as a defense mechanism. The bigger a sauropod grew, the harder it became for predators to threaten it. Yet, despite extensive study, questions remain about how their cardiovascular and respiratory systems supported such intense metabolic demands. Stegosaurs, with their distinctive plates and tail spikes, followed a slower, more measured developmental trajectory. Their bones show a shift from rapidly deposited fibrolamellar tissue to denser, more structured bone earlier in life than in other species. Unlike their fast-growing cousins, stegosaurs may have taken a more cautious path to maturity, perhaps due to differing environmental pressures or reproductive strategies. Did slower growth correlate with longer lifespans or greater parental investment? Or were these simply low-metabolism herbivores built for efficiency over speed? Arctic-dwelling dinosaurs like Pachyrhinosaurus perotorum offer another layer of complexity. Fossils from northern Alaska show that juveniles reached nearly half of their adult size within their first 2 years, an impressively fast start, likely a necessity in a region with limited growing seasons. But the growth slowed dramatically thereafter, suggesting a response to seasonal resource scarcity or extreme conditions. This seasonal pattern, recorded in the spacing of growth rings, poses a critical question: how did these cold-weather dinosaurs regulate their development and survive prolonged winters? Troodontids and other small theropods show a different trend: uniformly high growth rates paired with large brain cases and advanced sensory features. These creatures may have matured quickly to maximize survival in predator-dense ecosystems. Their relatively high metabolic rates, inferred from their bones and relatedness to birds, suggest warm-blooded physiology that required rapid growth just to sustain energy balance. But how these smaller dinosaurs managed their caloric needs during lean seasons or periods of migration remains unclear. Were they more bird-like in their endurance or closer to modern reptiles in energy conservation? Paleontologists have also uncovered intriguing evidence that even within a single species, growth rates could vary widely. In Maiasaura, the so-called good mother lizard, some individuals reached adult size in just 8 years, while others took much longer. This variation might reflect differences in access to food, climate conditions, or social rank. The concept of developmental plasticity, flexible growth based on environmental cues, may have been more widespread than previously thought. It's a behavior observed in modern reptiles and birds but rarely confirmed in extinct animals. Growth plates, the zones at the ends of bones where elongation occurs, further connect dinosaurs to modern birds. In many juvenile dinosaur fossils, these plates show signs of intense activity, reinforcing the idea that rapid growth was the ancestral state. The evolutionary inheritance of this feature speaks volumes. Birds, as living dinosaurs, still grow rapidly and reach maturity quickly, traits that may have originated over 200 million years ago. But the fossil record also hints that not all dinosaurs retained this growth style. Some, like certain ankylosaurs, seem to have developed more slowly, perhaps to conserve energy or reduce injury risk in their armor-clad bodies. Despite advances in bone histology, the reasons behind these diverse growth strategies remain mysterious. Was fast growth a defense against predators, a way to outcompete peers, or a reflection of high metabolism? Did slower growth equate to longer lifespans or more complex social structures? As more juvenile fossils are unearthed and scanning technologies improve, the timeline of dinosaur development will continue to evolve. But for now, growth rate variance stands as a clue-filled puzzle, offering a rare glimpse into how these ancient animals balance survival, size, and speed. Part 15. Soft Tissue Survival. The discovery of soft tissue remnants in dinosaur fossils has revolutionized our understanding of ancient life, challenging long-held beliefs about fossilization and organic preservation. In 2005, paleontologist Mary Schweitzer made headlines by identifying flexible blood vessels and cells in a 68-million-year-old Tyrannosaurus rex femur. This groundbreaking find suggested that under certain conditions, organic materials could survive far longer than previously thought. Since then, similar discoveries have been made in other dinosaur fossils, sparking debates and further research into the mechanisms behind such extraordinary preservation. One of the most remarkable specimens is Dakota, a nearly complete Edmontosaurus mummy discovered in North Dakota. This 67-million-year-old fossil retains not just bones but also skin impressions, muscle tissue, and tendons. Advanced imaging techniques, including CT scans, have allowed scientists to reconstruct its musculature and estimate its speed, revealing that it could run faster than previously believed. The preservation of such soft tissues provides a rare window into the biology and behavior of dinosaurs, offering insights that skeletal remains alone cannot. The preservation of soft tissues in fossils is a rare occurrence, as organic materials typically decompose over time. However, certain conditions can facilitate their survival. Rapid burial in anoxic environments, such as swamps or lake beds, can prevent decay by limiting exposure to oxygen and scavengers. Additionally, mineralization processes can replace organic materials with minerals, preserving the structure of tissues. These exceptional circumstances are necessary for the survival of soft tissues over millions of years. The molecular preservation of proteins like collagen in dinosaur fossils has been a subject of intense study. Collagen's fibrous structure and chemical stability make it more resistant to degradation, allowing it to persist longer than other proteins. Recent research has identified collagen in fossils over 80 million years old, suggesting that under specific conditions, proteins can survive deep time. These findings have profound implications for our understanding of molecular preservation and the potential for studying ancient biomolecules. The discovery of soft tissues in dinosaur fossils has also led to the development of new techniques in paleontology. Immunohistochemistry and mass spectrometry have enabled scientists to detect and analyze proteins and other biomolecules in ancient specimens. These methods have provided evidence of blood vessels, osteocytes, and even remnants of red blood cells in fossils, offering unprecedented insights into the cellular structures of extinct organisms. Such advancements are expanding the frontiers of paleobiology and molecular paleontology. Despite these advancements, the preservation of soft tissues in dinosaur fossils remains a topic of debate. Some researchers question the authenticity of these findings, suggesting that the structures identified may be the result of bacterial biofilms or other microbial activity. While these alternative explanations have been proposed, the majority of studies support the idea that under certain conditions, original soft tissues can be preserved over geological timescales. The ongoing research continues to refine our understanding of these processes. The implications of soft tissue preservation extend beyond paleontology. The ability to study ancient proteins and cells opens new avenues in evolutionary biology, allowing scientists to trace the development of physiological traits and behaviors in extinct species. Additionally, these discoveries have sparked interest in biotechnology, where ancient biomolecules could inspire the development of new materials and medical treatments. The intersection of paleontology and modern science is leading to innovative approaches in various fields. Looking forward, the study of soft tissue preservation in dinosaur fossils holds promise for further breakthroughs. Advancements in imaging technologies and molecular analysis techniques will likely uncover more about the biology of ancient life forms. As new fossils are discovered and analyzed, our understanding of the processes that allow for the preservation of soft tissues will continue to evolve. This ongoing research underscores the dynamic nature of science and our ever-expanding knowledge of the natural world. The survival of soft tissues in dinosaur fossils is a testament to the extraordinary conditions that can preserve life over millions of years. These rare finds not only challenge our understanding of fossilization, but also provide invaluable insights into the biology and evolution of ancient organisms. As research progresses, the mysteries of soft tissue preservation continue to captivate scientists and enthusiasts alike, offering a glimpse into the distant past that was once thought to be beyond our reach. Part 16. Pathologies and diseases. Fossilized bones often bear silent testimony to the injuries and ailments that plagued dinosaurs during their lifetimes. These pathologies, ranging from healed fractures to signs of infection, provide invaluable insights into the lives of these ancient creatures. One well-studied Allosaurus specimen exhibited numerous injuries, including broken ribs and infected bones, suggesting a life filled with physical challenges. The presence of healing in many of these injuries indicates that dinosaurs possessed robust immune systems capable of recovery, shedding light on their resilience and biological adaptability. Injuries were common among theropods, with many specimens displaying signs of trauma. Gorgosaurus, a close relative of Tyrannosaurus rex, has been discovered with healed limb fractures and skull wounds. The distribution of these injuries suggests frequent physical altercations, possibly related to hunting mishaps or social confrontations. Some fossils preserve bite marks that perfectly match the jaws of their own species, pointing to violent disputes among rivals. Whether over territory, mates, or dominance, these battles left permanent marks on their skeletons, a record of the brutal realities of prehistoric life. Dinosaurs did not only suffer trauma from external forces, they also endured internal maladies, including diseases still familiar today. Evidence of bone infections, such as osteomyelitis, has been found in several specimens. These infections would have caused intense pain and potentially limited mobility. Yet, the extent of healing seen in some fossils hints at a capacity for recovery. Some researchers suggest that the stress of surviving with disease may have shaped aspects of dinosaur behavior, encouraging group living or changes in feeding strategies to accommodate weaker individuals. Congenital deformities also made their mark in the fossil record. One striking example is a theropod that showed fused vertebrae, likely the result of a developmental disorder. Despite its condition, the dinosaur lived into adulthood. This survival raises compelling questions about whether some species had social structures that supported injured or disabled members, or whether such individuals adapted their roles within a group. The possibility of dinosaurs forming cooperative or at least tolerant social bonds becomes more plausible in light of these anomalies. Even some of the oldest dinosaur lineages show signs of cancer. A duck-billed dinosaur fossil revealed a growth consistent with an osteosarcoma, a malignant bone tumor. The very presence of such a disease in an ancient creature reshapes assumptions about the evolutionary roots of cancer. Rather than being a product of modern life, it appears that the susceptibility to certain diseases stretches deep into the past. Fossils like these become not just paleontological curiosities, but milestones in the timeline of disease itself. Sauropods, those massive long-necked giants, were not exempt from skeletal disorders. Some show signs of chronic stress or injury, particularly in their legs and hips, likely due to the strain of supporting such massive bodies. One specimen displayed oddly shaped limb bones, possibly due to overuse, injury, or genetic defects. These malformations offer a rare glimpse into the hidden cost of gigantism, revealing how biology strained to keep pace with evolutionary pressures that pushed body sizes to extreme limits. Smaller dinosaurs also bore the scars of hard lives. Dilophosaurus fossils have shown forelimb deformities, possibly from early life injuries or inherited conditions. For a predator relying on swift strikes and grasping prey, such limitations could be life-altering. Survival under these circumstances would have required behavioral adjustments, perhaps favoring ambush tactics or scavenging. These adaptations, etched into the fossil record, suggest a level of plasticity and resilience that contributed to dinosaur success across diverse environments. The accumulated data on dinosaur pathologies forms a vivid record of suffering, survival, and adaptation. From violent encounters with predators or rivals to slow degenerative conditions and infections, the fossilized remains reveal a full spectrum of physical hardship. Some species seemed more prone to certain injuries, hinting at behavioral or anatomical predispositions. Yet the overarching theme remains one of survival against adversity, a legacy of endurance that played out in silence beneath ancient skies. As imaging technologies and analytical methods improve, the study of ancient diseases is entering a renaissance. Modern scans reveal internal structures without damaging fossils, and microscopic analysis can trace the progress of infection or injury over time. Through these advancements, the hidden histories of dinosaur lives are being slowly restored. Each pathological specimen becomes a window into ancient ecosystems, not just a snapshot of death, but a story of how these creatures lived, suffered, and endured. Part 17. Camouflage and Patterning. The revelation that dinosaurs bore complex color patterns has upended long-held notions of drab gray leviathans roaming barren landscapes. Exceptional fossils preserving microscopic pigment structures, melanosomes, have unlocked the ability to infer hues and patterns with surprising precision. By examining the shape and arrangement of these melanosomes under powerful microscopes, scientists can distinguish between black, brown, reddish, and even iridescent coloration. This window into the Mesozoic palette transforms dinosaurs from monochrome silhouettes into vibrant living beings whose survival hinged on the interplay of color and environment. One of the most spectacular cases of dinosaur camouflage emerges from the nodosaurid Borealopelta, whose fossilized skin retains faint pigment residues revealing a counter-shaded pattern, a darker back contrasting with a lighter belly. Such coloration minimizes shadows and visual cues, making its bulky form harder to detect in dappled forest light. The precision of this pattern suggests a keen evolutionary pressure to blend seamlessly into its undergrowth habitat, hinting at stealth not normally associated with armored giants. Yet questions linger. Did Borealopelta alter its shades seasonally to match shifting foliage, or was its camouflage a fixed species-wide trait? Small ceratopsians like Psittacosaurus illustrate another camouflage strategy: disruptive patterning. Fossil evidence points to bold stripes and spots along its limbs and flanks, breaking up its outline against the mottled forest floor. Its dark facial markings may have served dual purposes, concealing its gaze from predators and conveying identity within herds. Whether Psittacosaurus juveniles bore different juvenile-only patterns for enhanced concealment remains unknown, leaving a tantalizing gap in understanding how these dinosaurs navigated the treacherous world from hatchling to adult. Feathered dinosaurs further enrich the tapestry of prehistoric coloration. Sinosauropteryx, one of the earliest known feathered species, sported a banded tail and raccoon-like facial mask, achieved through varying melanosome densities. This small theropod's orange and white counters shading likely provided both camouflage and visual communication across open habitats. Yet the degree to which its bands shifted with age or season remains a mystery. Did juvenile Sinosauropteryx look markedly different from adults, perhaps displaying brighter patterns to signal vulnerability and elicit parental care? In the canopy, Microraptor dazzled with iridescent plumage that caught sunlight in shifting rainbows. Its feathers contained flattened, plate-shaped melanosomes arranged to reflect specific wavelengths of light, creating an otherworldly sheen. Such iridescence could have played roles in courtship displays, species recognition, or territorial signaling among dense foliage. However, the prevalence of iridescent traits across dromaeosaurids and their exact behavioral contexts remain speculative, with few specimens preserving the delicate structures needed for confirmation. Moving from feathers to scales, the hadrosaur Edmontosaurus Dakota preserves exquisite skin impressions, showing scale mosaics of varying sizes and shapes. Patterns near its limbs hint at striping, possibly for camouflaging in floodplain settings, interspersed with shadows and grass. The subtle gradations in scale texture may have affected light reflection, serving to further obscure its massive form. Whether skin color in large herbivores responded to predator pressures or social signaling within herds continues to challenge paleobiologists seeking direct evidence. Beyond static camouflage, some theories propose dynamic color change in dinosaurs, akin to modern chameleons and cuttlefish. While no direct fossil evidence of chromatophores exists, the presence of specialized melanosome clusters in some specimens raises the possibility of rudimentary color shifts. Such abilities would have provided remarkable ecological advantages, temperature regulation, mood signaling, or rapid concealment. Until a fossil preserves skin layers showing these structures, the notion of color-shifting dinosaurs remains a tantalizing hypothesis. Paleocolor studies have also uncovered patterns hinting at sexual dichromatism, differences in coloration between males and females. Some feathered theropods show melanosome arrangements, suggesting brighter plumage on structures like crests or tail feathers. These accents could have been used for mate attraction or dominance displays, while the rest of the body remained camouflaged. Pinpointing true dichromatism in the fossil record, however, requires numerous well-preserved individuals of varying sex, a sample size that has yet to be achieved. The expanding field of dinosaur coloration research continues to challenge and enrich our vision of the Mesozoic world. Each new melanosome analysis brings us closer to reconstructing the living hues of creatures long gone, but also reveals fresh mysteries about the functions and dynamics of these colors. As techniques refine and more specimens emerge, the interplay of camouflage, display, and environmental adaptation in dinosaurs will be revealed in ever greater detail, painting the ancient past in living color once more. Part 18. Plate function. The iconic dorsal plates of stegosaurs have captivated scientists and the public alike, yet their true function remains shrouded in mystery. These tall, thin osteoderms, arranged in two staggered rows along the back, vary in size and shape among species, suggesting they played multiple roles. One prevailing theory posits that the plates acted as thermal radiators richly supplied with blood vessels. They could absorb heat from the sun in the morning and dissipate excess warmth as blood circulated through them. Yet recent studies of plate surface area and vascular density yield conflicting results, leaving open the question of whether temperature control was a primary function or merely a fortuitous byproduct of another adaptation. Beyond thermoregulation, display emerges as a compelling explanation. The sheer visibility of the plates, towering above the dinosaur's silhouette, would have made them ideal for species recognition and sexual signaling. In crowded forested environments, subtle differences in plate shape, size, and possibly coloration could have allowed individuals to identify conspecifics or assess mates from a distance. Some specimens even show evidence of grooves or scratches on the plate surfaces, hinting at possible adornment with keratinous sheaths or vibrant pigments. Whether these embellishments serve to attract mates, intimidate rivals, or establish social hierarchies remains one of the stegosaurian world's most tantalizing puzzles. Defense has also been proposed as a function, though its feasibility is debated. While the plates themselves are not robust enough to block bites or fend off predators directly, their imposing appearance could have made the animal look larger and more threatening, deterring attacks. In some modern herbivores, exaggerated structures serve as visual bluff. Stegosaurs might likewise have used their plates to create an illusion of bulk. However, no direct evidence such as healed bite marks on plates has yet been found to confirm this hypothesis, indicating that if the plates did serve in defense, it was likely psychological rather than physical. The orientation and spacing of the plates add another layer of complexity. Some reconstructions depict them leaning forward over the shoulders, potentially shading the neck and head during the midday sun. Others suggest a more upright alignment, maximizing exposure for display. Sediment compression makes reconstruction challenging, and variations among fossils suggest that plate orientation could have differed not just between species but between individuals, perhaps reflecting sexual dimorphism or age-related changes. This flexibility in arrangement underscores the difficulty in pinning down a single, universal function for the plates. Comparisons with other plated dinosaurs offer intriguing parallels and contrasts. Ankylosaurs, with their broad, flat dorsal scutes and heavy tail clubs, appear to have used armor primarily for defense, whereas stegosaurs retained light, translucent plates. Some have speculated that these differences reflect divergent ecological pressures. Ankylosaurs occupied low-browsing niches where physical defense was crucial, while stegosaurs, feeding on mid-height vegetation, invested in display and thermoregulation. Yet the evolutionary drivers behind these divergent strategies remain elusive, hinting at complex interactions between environment, behavior, and anatomy. Ontogenetic studies reveal that stegosaur plates changed dramatically as individuals matured. Juveniles bear small, simple osteoderms that gradually enlarge and alter in shape through successive growth stages. This transformation suggests that the plate's function evolved over an individual's lifetime. Young animals may have relied on cryptic behavior and rapid growth for protection, shifting toward display and thermoregulation as they reached adulthood. The precise timing and triggers of these ontogenetic shifts are still under investigation, offering a window into how developmental processes influenced stegosaur behavior and ecology. Advances in imaging techniques, such as high-resolution CT scanning, have begun to illuminate plate microstructure in unprecedented detail. Internal canals within the plates form complex networks, indicating potential for rapid blood flow. Yet the density and distribution of these canals vary widely among specimens, implying that vascularization and, by extension, plate function was not uniform across individuals or species. Deciphering whether these variations reflect environmental adaptations, individual health, or random developmental quirks remains a major challenge for paleobiologists. Seasonal and geographic differences may also have influenced plate function. Specimens from higher latitudes, where temperature fluctuations were more extreme, might show greater vascular investment in plates than those from equatorial regions. Isotope analyses of plate carbonates could someday reveal seasonal patterns of blood flow, shedding light on how these dinosaurs coped with changing climates. Until such geochemical studies are conducted, the role of plates in climate adaptation will continue to tantalize researchers. Ultimately, the true nature of stegosaur plate function may lie in a combination of roles rather than a single explanation. Multifunctional structures are commonplace in nature, and stegosaurs likely exploited their plates for thermoregulation, display, and deterrence to varying degrees throughout their lives and across environments. The challenge for modern science is to unravel these intertwined functions, reconstructing not only the anatomy but the dynamic lives of these remarkable armored giants. As new fossils emerge and analytical methods improve, the story of stegosaur plates will continue to evolve, bridging the past and the present in an intricate dance of form and function. Part 19. Spike and tail use. Among the most fearsome appendages of the Mesozoic world were the tail weapons of stegosaurs and ankylosaurs, evolving in parallel yet serving distinct functions. In stegosaurs, the thagomizer comprised 4 long, pointed spikes projecting from the tail's end, while ankylosaurs developed massive, bony clubs. Both designs hint at sophisticated defensive strategies, yet the degree to which they were used as offensive weapons, intraspecific combat tools, or display structures remains enigmatic. Fossilized trackways and bone beds sometimes place these armored dinosaurs alongside predators, but seldom in direct evidence of tail-driven violence, leaving paleontologists piecing together indirect clues from anatomy, biomechanics, and rare injury patterns. Stegosaur thagomizers exhibit remarkable variation in spike length, angle, and cross-sectional thickness across genera. In Stegosaurus stenops, spikes reach up to a meter in length, tapering to razor-sharp points. Biomechanical models suggest that a well-muscled stegosaur could swing its tail laterally with enough force to puncture the ribcage of an attacking Allosaurus. Yet the limited flexibility of the stegosaur tail, composed of stiff, interlocking vertebrae, raises questions about accuracy and reaction speed in real combat. Experimental reconstructions using robotic tails indicate that while powerful, the thagomizer's arc was slow, implying that stegosaurs may have relied on surprise or stationary posturing rather than rapid tail strikes. Ankylosaurid dinosaurs, by contrast, sported tail clubs formed by enlarged osteoderms fused at the tail's tip. These clubs, some weighing over 30 kilograms in large specimens like Ankylosaurus magniventris, acted as bludgeoning weapons capable of delivering devastating blows. High-resolution CT scans reveal internal zones of trabecular bone optimized to absorb impact, while muscle attachment scars along the caudal vertebrae indicate a robust musculature for rapid tail thrusts. Yet uncertainties persist. Did ankylosaurs strike downward at limb joints to immobilize predators, or deliver horizontal swings to break bones? The orientation of muscle scars provides hints, but fully fleshing out the motion range of the club remains an active area of research. The mechanics of tail movement, both in stegosaurs and ankylosaurs, depend on the interplay of vertebral articulation, muscle mass, and tendon elasticity. In stegosaurs, the proximal tail vertebrae show tall neural spines for anchoring epaxial muscles, but the distal vertebrae form a stiff whip-like structure that limits lateral motion. By contrast, ankylosaurs evolved increased joint mobility in middle caudal vertebrae, allowing wider swings of the club. Reconstructing the exact muscle arrangement relies on rare soft tissue impressions and comparisons with modern reptiles. Without direct evidence of muscle fiber orientation or tendon placement, scientists build three-dimensional models that offer plausible but unconfirmed views of tail dynamics. Evidence of tail-based combat emerges most directly from pathologies. Some theropod fossils show puncture wounds matching the dimensions of stegosaur spikes, suggesting that predators occasionally bore the brunt of a defensive strike. Likewise, ankylosaur vertebrae and limb bones sometimes display healed fractures consistent with blunt force trauma from a club. Yet the rarity of such injuries raises questions about their overall effectiveness. Were tail weapons primarily deterrents rather than reliable defenses? Alternatively, the absence of extensive damage in the fossil record could reflect rapid predation events that left little chance for a successful tail strike, or simply the low likelihood of precise strikes hitting vulnerable areas. Beyond defense, tails and spikes may have played roles in display and communication. Imagine a stegosaurus orienting its striped, or possibly brightly colored spikes in a coordinated display to establish territory or court mates. Ornamental keratin sheaths could have accentuated the spikes, making them visually striking at dawn or dusk. While direct pigment evidence is lacking for stegosaur plates and spikes, some researchers propose that vibrations produced by tail thumping against the ground or resonant osteoderm pulses in ankylosaurs served as seismic signals. Such acoustic communication, similar to tail banging in modern crocodilians, remains speculative but intriguing. Ontogenetic studies reveal that juveniles possess smaller, more gracile tail spikes or clubs, which enlarge and sometimes change shape as animals mature. In some stegosaurs, early growth stages show simple, cone-like osteoderms that elongate into full spikes over several years. Ankylosaur juveniles display knob-like tail tips that ossify and fuse into clubs only in late adolescence. These changes suggest that tail weapons serve different functions at different life stages. Perhaps juveniles relied on hiding and agility, while adults used tails for defense, social dominance, or mate competition once fully developed. Pathological evidence within the tail weapons themselves opens further mysteries. Several stegosaur spikes show signs of repair after breakage, with bone regrowth patterns indicating that broken spikes could regrow, albeit imperfectly. Similarly, ankylosaur clubs sometimes bear dents or microfractures in their cortical bone, smoothed over by healing. The frequency and distribution of such damage hint at individual variation in combat experience. Some adults display multiple healed spikes, suggesting they survived several predatory encounters, while others show pristine tail clubs, raising the possibility that certain individuals avoided conflict altogether. At the heart of these investigations lies the question of neurological control and behavioral context. Delivering an effective tail strike requires precise timing, powerful muscle activation, and spatial awareness to avoid self-injury. How did the dinosaur brain coordinate such complex motor tasks? Comparative studies with crocodilians and monitor lizards offer limited analogies. Fossilized endocasts of stegosaurs show relatively small cerebellar regions, areas linked to motor coordination, leading some to doubt highly targeted tail attacks. Ankylosaurs with slightly larger cerebellar volumes might have exhibited better tail control. Yet until live neural data emerges, the coordination of these weapons remains a profound mystery. The study of spike and tail use in dinosaurs exemplifies the synergy of fossil evidence, biomechanical modeling, and comparative anatomy. Each new discovery, whether a healed wound, a rare skin impression, or refined muscle attachment map, adds a brushstroke to a dynamic portrait of Mesozoic life. As 3D scanning, robotics, and paleoneurology advance, the dance of tail and weapon may finally reveal not just how these structures functioned mechanically, but how they shaped behavior, ecology, and the very survival strategies of some of Earth's most extraordinary creatures. Part 20. Flight Origins. The leap from ground-bound dinosaur to airborne avian stands as one of evolution's most dramatic transformations. Among the earliest clues are the long, asymmetric feathers of small theropods like Anchiornis huxley. Its four-winged anatomy with flight feathers on both forelimbs and hindlimbs suggests an evolutionary experiment in aerial control and gliding. Yet whether Anchiornis clambered into trees to launch or used its proto-wings to escape predators on the forest floor remains hotly debated. The absence of direct evidence for arboreal habits leaves paleontologists torn between the trees-down and ground-up hypotheses, each supported by skeletal nuances but lacking a decisive fossil that bridges the behavioral gap. In China's Liaoning province, Microraptor gui offers further insight into early flight mechanics. Its four-winged body appears optimized for gliding, long feathers on the arms, legs, and a feathered tail forming an airfoil reminiscent of a biplane. Wind tunnel experiments on life-sized models demonstrate that Microraptor could navigate steep descents, adjust its pitch by splaying toe feathers, and even perform controlled turns. Yet the creature's inability to generate sufficient lift for powered flight leaves it poised at the edge of avian evolution, a master glider that never quite mastered flapping flight. Understanding why such promising designs never transitioned fully to wing-driven locomotion remains a perplexing enigma. Closer to the ancestral line of modern birds lies Archaeopteryx lithographica, a late Jurassic icon blending reptilian and avian traits. Its robust forelimbs, asymmetrical primary feathers, and flexible wrist joints indicate a capacity for flapping flight, albeit weak and short-ranged. Debates swirl around whether Archaeopteryx launched from the ground with a wing-assisted incline running strategy, using its wings to scramble up inclines like a turkey, or whether it leapt from perches. The discovery of quill knobs on its ulna hints at powerful flight muscles, but without fossilized evidence of shoulder girdle soft tissues. Reconstructing its flight stroke remains speculative, shrouding its aerial prowess in mystery. Even before Archaeopteryx, the dragon-like Rhahonovis ostromi from Madagascar straddles the reptile-bird divide. Its robust shoulder articulation and well-developed muscle scars suggest a stronger flapping capability than its contemporaries. Yet its hips and legs retain terrestrial adaptations, implying a dual life of ground foraging and opportunistic flight. This mosaic of features blurs the linear narrative of flight evolution, raising questions about the sequence in which key adaptations, feathers, wings, keel-shaped sternum, emerged. Did powerful flight muscles evolve before full wing articulation, or vice versa? Without more complete remains of transitional species, the order of evolutionary innovations remains an open riddle. The function of early feathers adds another layer to the puzzle. Initial feather structures likely served for insulation or display long before aerodynamic use. Fossils of D-long paradoxus and other basal tyrannosauroids exhibit filamentous proto-feathers, suggesting insulation in warm-blooded dinosaurs. Yet the transition from simple filaments to complex veined feathers capable of creating lift is poorly understood. Did display-driven colorful feathers co-opt into aerodynamic surfaces, or did selective pressures for gliding drive the refinement of feather architecture? Pinpointing the selective pressures behind this morphological leap is crucial for mapping the trajectory from insulation to flight. Skeletal innovations accompanied feather evolution in concert. The shift from a solid, bony sternum to a keeled sternum, capable of anchoring large flight muscles, appears in late Cretaceous aviolans like Confucius ornis sanctus. Its fusion of hand bones and reduction of tail vertebrae signal advanced flight adaptations. Yet species like Geholornis prima retained long tails and clawed fingers, pointing to a spectrum of wing morphologies and flight styles. How selective forces sculpted this diversity, whether habitat constraints, predator avoidance, or foraging strategies, remains unclear. Each new specimen reshapes our understanding, but also deepens the mystery of flight's evolutionary drivers. Physiological demands of powered flight would have required high metabolic rates and efficient respiratory systems. Evidence of air sacs and hollow bones in non-avian theropods indicates that the pulmonary apparatus necessary for sustained flapping emerged before true birds. Yet the timing of these respiratory innovations relative to wing development is contested. Did complex air sac systems evolve to meet the oxygen demands of running dinosaurs first and later facilitate flight, or were they selected alongside early wing use? Unraveling these interdependencies is key to understanding how dinosaurs overcame the energetic barriers to powered flight. Modern birds employ intricate flight strokes and wing morphing to optimize efficiency. Tracing these capabilities to their dinosaurian roots poses another challenge. Joint structures in Maniraptorans reveal limited yet critical mobility in the shoulder and elbow, but without preserved soft tissues, reconstructing the range of motion is inferential. Recent discoveries of fossilized wing membranes in pterosaurs offer analogies but may mislead when applied to feathered dinosaurs. Achieving a biomechanical synthesis that accurately models the earliest flapping strokes remains a frontier of paleontological engineering. Ultimately, the origins of flight represent an evolutionary tapestry woven from multiple threads: feathers, skeletal modifications, respiratory changes, and behavioral shifts. Each new fossil discovery adds a patch of color, yet the seams between gliding and flapping, between ground run and tree launch, remain obscured. Unraveling this tapestry demands interdisciplinary collaboration, from aerodynamic modeling to developmental genetics. Only by integrating evidence across fields can the grand story of how dinosaurs first took to the skies be told in full, transforming mystery into understanding, one fossil at a time. Part 21. Swimming abilities. The notion of dinosaurs as exclusively land-bound giants has been dramatically overturned by discoveries suggesting some species were adept swimmers. Spinosaurids, most notably Spinosaurus aegyptiacus, exhibit a suite of adaptations for an aquatic lifestyle: paddle-like feet, a long, narrow snout filled with conical teeth, ideal for catching fish, and dense limb bones that would have reduced buoyancy. Yet the most striking revelation came with the unearthing of a deep, laterally compressed tail structure, resembling that of modern crocodilians, which likely provided powerful propulsion through water. How Spinosaurus balanced life between riverbanks and open streams, whether stalking fish in shallow currents or lurking in deeper channels, remains one of paleontology's most compelling questions. Beyond the giants, smaller theropods appear to have taken to water as well. Fossil remains of Baryonyx walker include partial gut contents containing fish scales, implying regular piscivory. Its forelimbs bear enlarged claws that could have served both to hook slippery prey and to aid in pushing against the riverbed. Stroke-based swimming, using alternating arm movements and undulating the torso, has been proposed, but without direct evidence of muscle attachments or soft tissues. The exact swimming style remains speculative. The interplay between its terrestrial running abilities and aquatic forays paints a picture of a versatile predator straddling two worlds. Hadrosaurs, the celebrated duck-billed dinosaurs, may also have been proficient swimmers. Their broad, flat tails and long muscular limbs suggest powerful strokes, while their lightweight skeletons would have aided buoyancy. Trackways found near ancient shorelines sometimes show parallel depressions, lacking foot impressions, interpreted by some as evidence of paddling tails dragging through soft substrate. Yet skeptics argue these depressions could result from subaqueous currents reworking sediment. The true swimming capabilities of hadrosaurs thus remain enigmatic, hinged on deciphering trackway taphonomy in ever-shifting floodplain settings. Sauropods, those colossal long-necked herbivores, present an even deeper mystery. Their immense size led early researchers to speculate they might have used buoyancy and water to support their weight. Fossilized limb bones of some sauropods show thickened cortical walls, a condition known as pachyosteosclerosis, which could counteract buoyancy and stabilize the animal in water. If sauropods did swim, or at least wade in deep rivers, their long necks and tails might have acted as natural balance beams. Yet the absence of unequivocal swim track fossils leaves open whether these giants truly entered deep water or simply forded rivers on strong limbs. Trackway evidence across multiple sites hints at dinosaur swimming behavior. In the early Cretaceous lacustrine deposits of South Korea, parallel grooves alongside intermittent footprints suggest that some ornithopods swam, using tail undulations as the primary propulsive force, while periodically touching the bottom with forelimbs. The patterns resemble those made by modern swimming reptiles in shallow water. However, alternative interpretations propose that these marks arose from subaqueous current draping over resting bodies. Untangling these possibilities demands meticulous sedimentological analysis, a task complicated by the hundreds of millions of years that separate us from these ancient waterways. Bone microstructure offers additional clues. Aquatic and semi-aquatic animals often develop dense bones to counteract buoyancy. Histological studies of spinosaurid and hadrosaur limb elements reveal increased bone compactness compared to strictly terrestrial theropods. This osteological signature suggests selection for subaqueous life, but quantifying the degree to which bone density contributed to swimming efficiency remains an active field of study. The relationship between bone architecture and locomotor ecology in extinct taxa teeters, between inference and experimentation, awaiting more refined biomechanical models. The sensory adaptations of swimming dinosaurs also spark curiosity. In modern aquatic predators, pressure-sensitive receptors along the snout detect water movements. Some spinosaurid fossils exhibit subtle channel-like grooves on the snout bones, raising the possibility that these dinosaurs possessed a sensory network akin to crocodilian integumentary sensory organs. If accurate, such features would have allowed spinosaurids to hunt blind in murky waters. Confirmation of these structures and their function, however, hinges on exceptionally preserved specimens capturing fine-scale cranial anatomy, which remain exceedingly rare. Reconstructing swimming behavior further relies on comparing dinosaurs with modern analogues. Crocodilians and aquatic birds provide models for limb stroke patterns and tail propulsion, yet fundamental anatomical differences caution against direct comparisons. Dinosaurs lacked the complex joint articulations of birds' wing bones and the tail vertebrae flexion range of crocodiles, forcing paleontologists to adapt modern frameworks judiciously. The challenge lies in building dynamic simulations that honor both the fossil evidence and the nuances of living animals, a multidisciplinary effort at the frontier of paleobiomechanics. Ultimately, the study of dinosaur swimming abilities unites sedimentology, bone histology, functional anatomy, and comparative ecology in pursuit of understanding how these creatures navigated ancient waterways. Each new fossil, from a fragmentary toe bone to an articulated tail, adds a pixel to the broader image of dinosaurs, not just as terrestrial titans, but as versatile denizens of land and water alike. As analytical techniques advance and fresh discoveries emerge, the mysteries of dinosaur swimming promise to yield surprising insights into the adaptability and ecological breadth of these iconic prehistoric animals. Part 22. Predator-prey tactics. The Mesozoic world was shaped by an endless arms race between hunters and the hunted, each innovation met with a countermeasure. The sheer diversity of strategies, ambush pursuit, group coordination and deceptive defense paints a picture of ecosystems teeming with tension and drama. Predators honed lethal adaptations, retractable claws for slashing, serrated teeth for ripping flesh, and powerful limbs for sprinting. Prey species evolved spikes, horns, armor, and herd behaviors to thwart attacks. Yet the fossil record offers only tantalizing snapshots, broken bones, healed wounds, and trackways frozen in time, leaving the true choreography of these deadly encounters tantalizingly out of reach. Ambush tactics appear in the anatomy of large theropods like Allosaurus and Dilophosaurus. Their robust hind limbs and flexible spines suggest bursts of speed and agility ideal for sudden lunges. Strike marks on prey fossils hint at attacks from concealed positions, along riverbanks or amid dense undergrowth. In coastal swamps, Spinosaurus may have lain nearly submerged, using its crocodile-like snout to snatch fish in a flash. Yet whether these giants lay in wait like modern crocodilians or prowled constantly in search of vulnerable prey remains uncertain, because the forests and waterways of the past conceal far more than they reveal. Pursuit predators relied on endurance and coordination. Smaller, feathered maniraptorans, such as Deinonychus, possessed long legs and stiffened tails, adaptations for high-speed chases. Some paleontologists argue these dinosaurs hunted in packs, isolating weak or injured individuals from herds of larger herbivores. Fossil sites containing multiple individuals alongside Protoceratops skeletons support this view, but critics point to alternative explanations: social scavenging or chance accumulation. Until bone beds preserve definitive evidence of cooperative killing strikes, the debate over dinosaur pack hunting will continue to fuel controversy. Even solitary predators may have employed tactics more complex than mere chase and ambush. Modern birds of prey use surprise and aerial agility. Similarly, smaller dromaeosaurs with feathered forelimbs could have used short bursts of wing-assisted hops to navigate terrain or corral prey. The degree to which proto-wings contributed to hunting success remains speculative. Fossil footprints preserving parallel tracks of predator and prey hint at strategic cornering near embankments or cliff edges, yet without soft tissue preservation, reconstructing their precise movements remains a tantalizing puzzle. Prey animals countered with an arsenal of defensive behaviors and anatomies. Herds of hadrosaurs scanned the horizon for threats, relying on collective vigilance and rapid flight in all directions at the first sign of danger. Ceratopsians formed protective circles, horns bristling at the center, while stegosaurs turned tail spikes outward in a living mace formation. In some bone beds, juveniles cluster at the core of the group, shielded by adults, a behavior seen in modern ungulates. But whether such formations were instinctive or coached socially is unknown, leaving the nuanced dynamics of herd defense shrouded in mystery. Bone pathologies reveal the scars of these battles. Triceratops skulls bear healed puncture wounds, matching the curvature of Tyrannosaurus teeth, indicating survivors of fierce clashes. Allosaurus vertebrae sometimes show gouges from stegosaur tail spikes, their distribution suggesting opportunistic strikes at flanks during skirmishes. The spacing and angles of these injuries can hint at attack strategies, high versus low, lateral versus frontal, but interpreting these angles demands careful 3D modeling, a frontier only now being explored. Each healed injury is a testament to survival, but also a clue to the lethal tactics employed. Predators relied on more than brute force. Sensory adaptations sharpened hunting proficiency. Binocular vision in many theropods allowed depth perception critical for gauging strike distance, while large olfactory bulbs in Tyrannosaurus imply an acute sense of smell for detecting carcasses or hidden prey. Some Spanosaurid snouts feature pits that may have housed pressure-sensitive receptors, enabling underwater detection of ripples similar to modern crocodilians. Confirming these soft tissue structures in fossils is rare, yet their possible existence opens new vistas on how dinosaurs sensed and tracked their quarry. Scavenging blurred the line between predator and prey tactics. T. rex, long portrayed as an apex hunter, likely scavenged as readily as it hunted, its massive bite force capable of crushing bone and competing with smaller carnivores for carrion. Toothwear patterns and coprolites rich in indigestible fragments suggest a diet that included bone marrow, implying that even formidable predators maximized resources. The extent to which dinosaurs targeted live prey versus scavenged remains a key question in understanding their ecological roles and energy budgets. The fossil record's bias toward hard parts and catastrophic bone beds leaves many tactics hidden in soft tissues and fleeting behaviors. Trackways may capture a chase, but rarely the kill. Pathologies record injuries but not the circumstances of their infliction. Experimental biomechanics, advanced imaging, and comparison with living analogs provide valuable insights. Yet the true fluidity of predator-prey interactions, split-second decisions, cooperative maneuvers, and adaptive learning remains one of paleontology's most captivating enigmas. Each new discovery brings us closer to reconstructing these ancient battles, yet also reminds us that much of this epic drama unfolded just beyond the reach of fossilization. Part 23. Bone Bed Enigmas. Fossil bone beds serve as frozen tableaux of ancient lives, capturing moments of mortality on a grand scale. Vast accumulations of skeletons, sometimes numbering in the thousands, offer glimpses into catastrophic events, social behaviors, and ecological catastrophes. Yet interpreting these dense assemblages is fraught with uncertainty. Were these dinosaurs victims of floods, drought-driven die-offs, volcanic ash falls, or social gatherings gone wrong? Each bed demands careful disentangling of transport, burial processes, and biological signals to reconstruct the drama behind the stones. In the floodplain mud of Montana, the nesting colony of Maiasaura presents one of the most compelling bone bed puzzles. Hundreds of juveniles and adults lie mixed together in a layer less than a meter thick, suggesting a rapid burial, perhaps by a sudden crevasse splay or flash flood. The tight clustering of nests raises questions about colony dynamics. Did the herd return year after year to the same site, only to suffer repeated natural disasters? Or does the bone bed represent a single generation overwhelmed by an unprecedented climatic event? Disentangling repetitive seasonal layering from one catastrophic horizon remains a central challenge. Alberta's plains reveal massive Centrosaurus bone beds, where tens of thousands of horned dinosaurs appear to have perished simultaneously. Some theories propose drought-induced congregation at dwindling waterholes, followed by mass mortality and scavenger-mediated scattering of bones. Yet other researchers see evidence of riverine transport, bones sorted by size, snout-first orientations, and aligned long bones hint at currents carrying carcasses downstream. Determining whether Centrosaurus gathered in life or only in death continues to spark debate. Carnivorous bone beds are rarer but equally enigmatic. A quarry in Utah yielded multiple Allosaurus individuals tangled with sauropod remains, suggesting either a fatal ambush by prey fighting back or a feeding frenzy that turned disastrous. Broken teeth embedded in limb bones and grooves on vertebrae from theropod bites hint at intraspecific scavenging. Did these predators converge on a single carcass until collapse, or did a pack of hunters attack together only to be overwhelmed by a massive sauropod's final desperate defense? The evidence remains tantalizingly ambiguous. In Patagonia, the tiny sauropodomorph Mosaurus appears in age-segregated bone beds, with dozens of juveniles preserved together, separate from adults. This pattern suggests nursery herds, where young congregated for safety or social learning. Yet adult skeletons lie kilometers away, implying seasonal fission, fusion dynamics, or periodic migrations. Whether juveniles remained in stationary creches and adults ranged more widely, or whether differential preservation simply selected for discrete juvenile sites, is a puzzle that intertwines behavior with taphonomy. Even within a single bone bed, taphonomic signatures vary dramatically. Some fossil pits show pristine articulation, indicating rapid burial in life position. Others contain scattered, disarticulated bones sorted by water currents. This patchwork reflects fluctuating depositional environments, shifting channels, mud flows, and drought-cracked surfaces. Piecing together a comprehensive burial history demands integrating sedimentology, bone orientation studies, and microstratigraphic analysis to reveal the sequence of events that converged on each site. Time averaging adds another layer of complexity. A bone bed may incorporate remains spanning hundreds or even thousands of years, blurring the line between a single mass death and a palimpsest of smaller events. Radiometric dating and careful examination of fossil weathering stages can distinguish episodic accumulation from instantaneous catastrophe. But few sites yield the high-resolution chronology needed to resolve these intervals. Consequently, some bone beds resist straightforward classification as either catastrophic or attritional assemblages. Microvertebrate bone beds, concentrations of teeth, small bones and fish scales, present their own enigmas. Often formed in riverine eddies or at the margins of drying ponds, these beds trap the smallest denizens of ancient ecosystems. Yet the ecological signals are muddled. Seasonal die-offs of amphibians might overlap with mass fish kills, and storms can inundate nesting ground corners, mixing terrestrial and aquatic remains. Interpreting these microassemblages demands fine-scale taphonomic work and experimental analogues to tease apart the drivers of each concentration. Ultimately, bone beds remain both windows and mirrors, windows into vanished worlds and mirrors reflecting the biases and limits of the fossil record. Each quarry presents a unique combination of anatomical, geological, and ecological factors, defying one-size-fits-all explanations. As high-resolution imaging, geochemical fingerprinting, and refined dating methods advance, the hidden narratives within these dense fossil deposits will slowly emerge. Until then, bone bed enigmas stand as monumental reminders of the intricate interplay between life, death, and the forces that bind them in stone. Part 24. Extinction Cause. The end of the Cretaceous period, roughly 66 million years ago, marks one of the most dramatic mass extinctions in Earth's history. A global layer enriched in iridium, a metal rare on the planet's surface but abundant in asteroids, signals the arrival of a cataclysmic bolide. Impacts spherules, shocked quartz grains, and a 180-kilometer-wide crater at Chicxulub in modern-day Mexico all point to a single, momentous collision. The kinetic energy unleashed would have triggered mega-tsunamis, global wildfires, and an impact winter as soot and dust shrouded the atmosphere. Yet questions linger about the precise sequence and duration of these effects, and whether a single strike was sufficient to topple the non-avian dinosaurs and countless other species. In parallel, massive volcanic eruptions were pouring basalt across what is now western India, forming the Deccan Traps. These eruptions, extending before and after the Chicxulub impact, released prodigious quantities of sulfur dioxide, carbon dioxide, and other volatiles, potentially driving episodes of intense climate warming, acid rain, and ozone depletion. Some models suggest that peak volcanism coincided closely with the impact, compounding the environmental stress and hindering recovery. But disentangling the relative contributions of impact and volcanism to the extinction event remains a contentious challenge, with geochronologists striving to refine eruption timelines to within tens of thousands of years. Immediate aftermath of the impact likely saw global temperatures plummet as sunlight dimmed under an aerosol veil. Photosynthesis stalled, collapsing food chains from plankton in the oceans to flowering plants on land. Yet sediment records reveal a brief fern spike, where hardy ferns proliferated in the ash-covered landscapes before giving way to more complex flora. How long these dark, cold conditions persisted, and to what extent they differed between hemispheres, remains debated. Some data indicate a return to near-normal light levels within months, challenging early notions of a multi-year impact winter. Oceans too would have felt the shock. Rapid acidification from dissolved carbon dioxide and sulfuric compounds, combined with depletion of photosynthesizing plankton, likely created widespread dead zones. Marine reptiles and ammonites vanished, while survivors like sharks and bony fishes adapted or retreated to refugia. But precise patterns of marine extinction vary by region and depth, sparking questions about the role of habitat specialization and ocean circulation in determining which lineages persisted. The selective mortality among terrestrial vertebrates offers further puzzles. Non-avian dinosaurs disappeared entirely, yet small nocturnal mammals and archosaurs, precursors to modern birds and crocodilians, survived. Body size appears critical. Smaller animals required fewer resources and could shelter in burrows or aquatic hideouts. Yet even among small species, survival was uneven, hinting at behavioral and physiological factors such as metabolic flexibility or dietary generalism that conferred resilience. Pinpointing these traits in extinct creatures without living analogues forces researchers to infer from teeth wear, bone microstructure, and burrow fossils. Environmental stresses continued for millennia. Greenhouse warming from volcanic CO5 emissions may have followed the initial cooling, creating a roller coaster climate. Acid rain from sulfuric aerosols would have leached soils, impacting plant recolonization and nutrient cycles. Freshwater ecosystems dependent on pH stability would have struggled to recover. The combined oscillations of cold and heat, acidity and alkalinity present a climatic seesaw whose effects on evolving ecosystems are still being reconstructed through sediment cores and isotopic analyses. Recent studies suggest the extinction event was not a singular flashpoint, but a window of elevated extinction risk lasting several 100,000 years. In some locales, faunal turnover appears gradual, with species disappearing before the iridium layer. These patterns imply that Earth's biosphere may have been under stress from long-term environmental change, with the impact serving as the coup de grace rather than the sole cause. Identifying early warning signs of decline in the fossil record could reshape our understanding of how ecosystems unravel under protracted stress. The extinction's legacy resonates today, offering analogies to modern biodiversity crises. Rapid climate change, habitat loss, and pollution mirror ancient pressures of warming, acidification, and ecosystem collapse. By studying the end Cretaceous events' complex interplay, asteroid fallout, flood basalt eruptions, climatic oscillations, and biotic selectivity, scientists hope to glean lessons on resilience, tipping points, and recovery. Yet the deep past holds enigmas that continue to challenge assumptions, reminding us that Earth's most profound upheavals often emerge from the convergence of multiple cataclysms. Ultimately, the cause of the dinosaurs' demise remains a tapestry woven from cosmic and terrestrial threads. While the Chicxulub impact provides a vivid focal point, the broader context of volcanic might, climatic upheaval, and ecological vulnerability paints a richer, more intricate picture. As high-precision dating, advanced geochemical proxies, and refined climate models converge, the extinction's final portrait emerges in ever sharper relief, yet always with margins of mystery that beckon further discovery. Part 25. Giant Egg Enigma. Among the most awe-inspiring discoveries in dinosaur paleontology are the colossal eggs unearthed in Argentina's Orca Mahuevo nesting grounds. Measuring up to 40 centimeters in length and nearly 30 centimeters in diameter, these eggs eclipse those of any modern land animal. Their sheer size conjures images of embryonic giants developing within thickened shells, gestating for months beneath layers of sediment. Yet it was only in the late 1990s that these enormous clutches were recognized, hidden within volcanic ash and mudstone for some 80 million years. The realization that creatures capable of laying such massive eggs once roamed the Earth reshaped assumptions about dinosaur reproduction and physiology. How such large eggs achieved successful hatching remains one of the field's most tantalizing riddles. Assigning these giant eggs to a specific dinosaur species has proven remarkably difficult. Although titanosaur embryos have been found in smaller eggs from the same sites, no embryonic remains have yet been discovered within the largest eggs. This absence raises the possibility that an entirely different, perhaps yet to be identified, sauropod lineage laid the biggest eggs. Tooth and bone fragments scattered among the nests offer tantalizing hints, but the fragments are too incomplete to confirm identity. As a result, paleontologists debate whether the giant eggs belong to a known titanosaur or represent a ghost lineage known only through its reproductive trace. Resolving this mystery could illuminate evolutionary branches of sauropods that vanished without leaving adult skeletons behind. The microstructure of giant eggshells presents its own conundrums. Detailed analysis reveals thick, multi-layered shell units with tightly packed pores, an architecture that must balance strength with the need for gas exchange. If the pores are too small or few, embryos risk suffocation, too many or too large, and the shell loses integrity. Yet in these enormous eggs, the pores are both abundant and diminutive, suggesting a highly specialized system for oxygen uptake and carbon dioxide release. How such extreme eggshell designs evolved, and what physiological adaptations embryos possess to survive within them, remains an open question that challenges our understanding of dinosaur developmental biology. Nest architecture surrounding these giant eggs hints at complex reproductive behavior. In some clutches, eggs are arranged in concentric rings, while others appear randomly dispersed. Evidence of plant debris and soil clumps between eggs suggests deliberate layering, possibly to regulate temperature and moisture. The absence of clear nests in certain layers implies that some eggs were buried under thick sediment, washes rather than neatly positioned by the mother. Whether these variations reflect different species' nesting strategies, environmental constraints, or seasonal shifts, the patterns raise questions about how giant egg layers managed incubation challenges at a continental scale. Embryonic development within giant eggs is poorly known, yet the few embryos recovered from smaller titanosaurs exhibit advanced ossification and possible oxygen scavenging adaptations. In smaller eggs, embryos appear robust, with well-formed limbs and skulls predating hatching by weeks. Extrapolating to much larger eggs suggests that hatchlings of giant eggs would have emerged vulnerable to collapse under their own weight if development proceeded at the same pace. Some researchers propose prolonged incubation periods, allowing gradual ossification and strength building. Others suggest hatchlings may have been semi-precocial, able to wriggle into shallower sediment or water to ease emergence. Unraveling these developmental dynamics hinges on finding the first giant egg embryos. Variation in egg size within single clutches adds another layer of mystery. Some nests contain a mix of giant and more modestly sized eggs, leading to speculation that females of different ages or sizes laid clutches simultaneously. Alternatively, mixed clutches could result from communal nesting by multiple species, each using the same prime nesting grounds. If true, these sites would represent dinosaur supercolonies, where diverse sauropods synchronized breeding to overwhelm predators and optimize habitat use. Confirming or refuting these scenarios requires mapping egg size distributions across multiple quarries and correlating them with trace fossils of adult dinosaurs. The incubation demands of giant eggs likely imposed significant energy costs on parent dinosaurs. Maintaining optimal temperature and humidity over an egg mass that could weigh tens of kilograms would require continuous nest attendance or ingenious environmental exploitation. Some have likened potential strategies to modern megapods, birds that bury eggs in geothermal soils or compost heaps to regulate heat. Whether dinosaurs used similar geothermal features, volatile-rich soils, or simple communal huddling remains unknown. Uncovering ancient geothermal vents or plant-rich nesting mounds in fossil strata could provide the first clues to these prehistoric incubation methods. Evolutionary drivers behind the emergence of giant eggs remain speculative. Larger egg size can confer advantages. More yolk reserves yield well-developed hatchlings, reducing early mortality. Conversely, producing massive eggs limits clutch size, risking population stability if few offspring survive. The balance between these conflicting pressures likely shaped reproductive strategies in giant egg layers. Did selective pressures favor fewer but more vigorous offspring in challenging environments, or did egg gigantism serve as a byproduct of extreme body sizes in some sauropods? Illuminating these evolutionary trade-offs requires integrating data on egg size, body mass estimates, and climatic reconstructions across the late Cretaceous. Ultimately, the giant egg enigma stands as a testament to the gaps in our knowledge of dinosaur life history. These monumental eggs encapsulate questions about species identity, embryonic physiology, nest construction, and evolutionary strategy, each layer of mystery inviting new discoveries. As prospective sites in South America and Asia yield fresh clutches, and as imaging technologies uncover hidden embryos, the secrets of these colossal eggs may finally crack open. Until then, the giant egg remains an enduring symbol of prehistoric wonder, challenging paleontologists to imagine the incredible orchestration required to hatch a sauropod behemoth from an egg larger than any laid by a land animal today.